Yes.
What should the chat?
Yeah, because we said 5:34, welcome registration meeting. Okay, but then they just say, yeah.
Okay, I can accept them all, or
Yeah.
wait.
No, in order to get the message, you have to accept.
I'll wait.
Then we can send it from the chat.
Yep.
Just what you want to say? Presentation will start.
Yeah, you want to start?
Disasters.
Good, how are you?
Good, yeah. Hey.
Je vais avoir une dinguerie ici, la cuisine de Bassi.
So I walked to the train station. Yeah, there's a Wi-Fi. Oh, there's a gas Wi-Fi. Yeah. I'm searching for every room.
En fait, c'est ce que tu dis là avec la petite poupée qui se trouve à la base de la caméra. C'est que quand nous on parle ici de la salle. De toute manière, moi je privilégie toujours ce qui vous revient. Oui, bien sûr.
Thank you so much.
C'est la musique. Dans ce cas-là, moi je déconnecte de celui-ci, et puis on voit ça, je fais de l'intégration ça, on fait un mix propre. On va aller, ouais, on va.
No, can we have the controller? How the speaker can see it?
They can see it.
They see it.
Okay, this one.
Okay,
Yeah.
And then
If you click it, it will change here. So this is.
okay,
Yeah.
so change the slides.
Yeah.
Forward and backward. Okay. What is the
Du coup, ça devient un SDA direct. Ouais. En 5 mètres. Par contre, je peux pas avoir un preview sur ce que je fais. Ou alors on utilise du coup le dernier convertisseur. Ah ben non, je peux trop. Du coup, ça, mode changé. Il faut le micro, je vais le mettre là-dessus. Ah ben oui, le téléphone. Pour le rec, on va pas trop non plus. Ça, c'est pas le bon. Ça, c'est le. Ah oui, c'est bon. Parce que du coup, on va avoir besoin. Ok, donc ça c'est IG, SDI. De SDI. Il faut que de SDI, c'est pas là. Moi j'en ai un. Mais normalement j'en avais pris en plus. Je suis d'accord là. Moi j'ai jamais vu. Par contre, j'ai pas je dois râler le métro, c'était une c'est pour l'installation. J'avais plus ah mais oui. Ça marche pas tout bien. Ah, il y a un micro ouvert oui, un micro-main, je pense.
Blah, blah, blah. Okay. Blah, blah, blah. Okay. Ok, we can... dov'è Hervé adesso? È lì. Okay, here we go. Hoping that everything works. So hello everybody, welcome to the GENE-SWitCH final conference. It's fine? Okay, so welcome to the GENE-SWitCH final conference. I'm Elisabetta Giuffra, the coordinator of the project. We'll be here Hervé o'clock with deputy coordinator. And we are going to start the first session at 4 p.m. There will be 3 talks by Alan Archibald as introductory talk, Hervé o'clock, Sylvain Foissac and Sarah Diabali, and Martijn Derks, before the coffee break. During this time, please write your questions in the chat, because then at 18:10, we will have the discussion, the Q&A session, and we will assemble in this time your questions and we will discuss them. So I give the floor already to Alan, who is connected. It's 2 minutes in advance, but it should be fine. Can you confirm that is going to? Oh, okay. Hi, Alan.
Hi.
The floor is yours. Thank you very much.
Right, if I can find the right bit.
We cannot hear you.
Okay, let me take that out of there. Can you now see my screen?
Yes.
Okay.
Yes.
Yes.
Okay, does that change the slide?
Yes.
Yes.
Yes, okay, right. Thank you for the opportunity to join the conference online. Makes it much easier for me. So I'm going to introduce this first session, starting off by observing that richly annotated reference genomes are absolutely critical resources, shared open access resources for 21st century biological research. They're also incredibly valuable for effective exploitation, for example, for genomics-enabled prediction, i.e. for selective animal breeding. Now, these are, or have been certainly, expensive resources to develop, and so international collaborative consortia, such as GENE-SWitCH and Eurofang and the other players, have been critical in pursuing this. So how did we get here? Well, it was 20 odd years after Watson and Crick discovered the structure of DNA before a genome was sequenced. So Fred Sanger's group, including a graduate colleague of mine, sequenced a bacteriophage back in 1978. And the speed of sequencing developed rapidly after that, with the Human Genome Project being launched in 1990, and the first 2 European farm animal projects, PigMap and ChickMap, being launched later in the 90s. And by the time we get to the opening of the new century, there was already a draft human genome available to everybody. And about that time, animal breeders launched the notion of using genomic information for selective animal breeding and developed the theory of genomic selection. And into the first decade of the new century, we had genome sequences emerging for mouse and cattle and horse. And the human genome apparently was finished for the first time of many times in 2003, a project that cost $3 billion to get to that stage. And a year later, the first of our two GENE-SWitCH species, the chicken, or strictly speaking, the red jungle fowl, was sequenced and published in Nature. And then in 2012, the second of our GENE-SWitCH species, the pig, that sequence was released. So we have this string of bases in the terms of the content of an organism's genome, strings of A, G, C's and T's. But how on earth do you read that? How on earth do you make sense of it? Which are the bits that are providing the important instructions? Well, in essence, there have been two approaches to discovering functional sequences. One's an evolutionary approach, taking the view that sequences that are conserved across many organisms and species must be important. So you can discover which sequences are conserved by comparing the sequences of many species, but what is that conserved element? What does it do? And of course, just because something is highly variable across different organisms doesn't mean it's non-functional. And if one thinks particularly of immune genes, then that's pretty critical. The other approach is to take a functional or biochemical approach to develop things called assays by sequence. And the pioneers of that on a genome scale was the ENCODE project, and with subsequent other human-centric ones in terms of IHEC and the epigenome roadmap. And FANG appears in that context. This approach, however, is incredibly expensive, and you're now exploring a four-dimensional space of location plus time. And then trying to sort out from these biochemical reactions what's biologically meaningful and what's noise. So the Human ENCODE project published its first comprehensive survey of DNA elements in the human genome in 2012. And the conclusions from that compendium of papers was that 80% of the genome sequence participated in at least one biochemical reaction. Were those meaningful, or were some of those just noise? Another key observation was that promoter functionality can explain most of the variation in gene expression. And critically, from the perspective of selective breeding or dissecting genetic predisposition to disease, variants associated with SNP in genome-wide studies are enriched within non-coding functional elements, so the bits that don't encode the sequences of the proteins. So if we pick up a timeline and remember that in 2012 the pig genome was sequenced, but that was also when the human community published their ENCODE compendium. A number of other genomes got sequenced in that second decade of the century, and then about halfway through the decade we see the launch of the Functional Annotation of Genomes initiative, and a concept that had been conceived back in 2012 at meetings between the European Union and US groups in the Animal Biotechnology Working Group. And then in 2016, a critical meeting was held when the EU looked at the projects it had funded in farm animal genomics and breeding over the preceding years and posed the question, what next? And a year after that workshop, in which Elisabetta and myself and others made the case for an ENCODE project for farmed animal species, a call was launched by the European Commission for such projects for agriculturally and aquaculturally important species. And then in 2019, these three Horizon 2020 FANG or AquaFANG projects were launched. And the one we're focusing on today is GENE-SWitCH. But there are parallel efforts going on in AquaFANG and BobReg. And the sum of money invested in these three projects together is 18 million euros, but that's for 9 species, whereas the Human ENCODE project for a single species. The one, of course, that we're most interested in ourselves cost $250 million. So as well as moving towards functional annotation of these genomes, there were improvements in the pig genome, and we eventually got sequences of genuine chickens, one a broiler and one a layer. And looking forward, we're looking to genome sequences that cover the complete genomes from one telomere to the next. And thinking ahead to having genome resources that can capture genomes of multiple individuals in order to get a better view of the genomic resources of a species. So we come to functional annotation of animal genomes. A number of assays are used to explore this, and Hervé will explain these in greater detail shortly. So we have capture high C that concerns interactions between the likes of promoters and other elements such as enhancers. Attack seek that allows us to look at areas of the chromatin that are open, and an assumption that those are bits that are active at any given time. ChIP-seq assays for histone modifications, again, that point to different regulatory elements. Reduced representation bisulfide sequencing and whole genome bisulfide sequencing allows us to identify modified bases, particularly, or specifically rather, methylated bases. And looking at outputs from the genome in terms of transcripts, both messenger RNA transcripts, small RNA, and ISO-seq's a way of looking at the messenger RNA, but in a more comprehensive way that allows you to see complete transcripts. And combining the information from these assays, one can identify key elements such as promoters and long-range regulatory elements. But we're doing this not for a single time point or for a single species in GENE-SWitCH. We're doing it across developmental times, from early organogenesis through to newborn and hatched individuals. Looking at the way in which different elements are switched on or switch off between these developmental stages, and so giving us a more comprehensive view of the functional elements within a genome. And so the talks you're going to hear this afternoon or this morning, depending which continent you're on, concern pillar one of the project. So this is functional annotation across tissues and developmental stages, work packages 1 through to 3. But these are key elements plugging into the likes of pillar 2, looking at ways in which this information can be used for precision animal breeding. Now, as I mentioned, the European Commission are pouring 18 million into these 3 projects. Why are they doing it? Well, the why they're doing it is that genomics-enabled animal genetic improvement has delivered already socioeconomic impact that is measurable. It was implemented in terms of genomic selection in the first round within 7 to 8 years of the development of the theory, and can be implemented with 1 to 2 years of the availability of the refined genomic information. So the information we and others are generating in these FANG projects will allow rapid refinement and improvement of genomic selection and selective breeding going forward. So that will allow us to address societal changes, including the demand for animal protein, the need to meet that demand in more efficient and sustainable manners, and being able to conduct selective breeding in an agile and accurate way, allowing this early selection of parents of future generations based on molecular genotypes, and changing those selection indices increasingly by our understanding of the relationship between genotype and phenotype, and being able to do that in response to a variety of challenges, including, of course, climate change. And these tools allow us to manage genetic resources that are key to animal productivity across the planet. And I think I'll now stop sharing and hand you over to the first of our speakers who will tell us about the results from the project, Hervé Alcott.
Thank you, Alan. I will follow with my presentations, which is based on the collection of samples we made during the project and the assays by sequence we provided to the scientific community. So to come back to the introduction by Alan, the idea to work on developmental stages is really to give more information on the way conserved regulatory elements are important during the formation of vertebrate structures and body, but also what is specific to each species and how these specificities can also be interpreted by looking at the effect of genetic and environmental interactions during the formation of organs and how it can impact the adult phenotype. So this is something that has been shown during early development in pigs, where you can see that the gene expression on the right and the chromatin accessibility on the left with the ataxic data shows a similar dynamic during development. And you can see that how cells are expressing genes and how the chromatin's accessibility is changing during time is correlated in this tissue for this species. And how our project was to look at this, but in different tissues and in different stages of development. So we decided to work on two species, the chicken and the pig. And by comparing 3 phases of development, so the early organogenesis, so chicken embryo at 7 days of development and a pig embryos at 30 days of development. The late organogenesis, so by looking at the chicken embryos of 15 days of development, so the start of the second third of the embryonic development, and pig embryos at 70 days of development. And finally, we looked at our mature organs in newborns by comparing chick and piglets of 1 day. And we also sampled different tissues with a focus on 7 main tissues, which are the liver, the lungs, and the small intestine, representing the endoderm, the kidney and the skeletal muscle to represent the mesoderm layer, and the skin and the cerebellum to represent the ectodermal layer. So we are trying to sample tissues from the different embryonic layers origins, and to see what is conserved also due to this early common origin for these different tissues. So this is a bit a summary of what we did. So for the pig, we use the large white breed, and for the chicken, we use the advanced intercross line that has been developed at the Roslin Institute. And for each stage, we use 4 replicates, 2 males and 2 females. And for the early stages, so either the chicken embryos or the fetus of 30 days, we worked with pools because it was not possible to only work on individuals because we have a small amount of biological materials. We have done additional biobanking for 6 tissues that are the gonads, the cortex, the cerebral cortex, the large intestine, the heart, the stomach, and the spleen. And for each tissue and organisms, we made metadata that have been stored on the Fang data portal and the biosamples database. We also deposit all the protocols that we use for the tissue samplings, for the DNA and RNA extractions, and all this information is publicly available on the Fang data portal and on the biosamples database that is based at the EBI center. So this is just an example of the classical metadata you can get when you go to the biosamples database. So you have the identifiers, and then you can see you can have the birthplace, the birth weight, the breed, the sex of the animal, the mother ID, the father ID, and all this information that is really important to be able to reuse the data that will be produced from these tissues and biological samples. Then we move to the molecular assay. So using these tissues, we decided to produce different data sets that Alan presented briefly before. So we have some that are looking at chromatin conformations, like promoter capture IC, ChIP-seq and CTCF proteins, chromatin accessibility by using the ATAC-seq, chromatin marks by ChIP-seq, looking at 4 different chromatin marks that are H3K4 monomethylated for enhancers, H3K4 3-methylated for promoters, H3K27 3-methylated for bivalent marks and repressive regions, and H3K27 acetylated for active transcription. We also looked at DNA methylation by using bisulfite sequencing, either at the whole genome level or by using capture of CpG island using RRBS. And we also done a huge transcriptomic repertoire for all the transcripts we can get for each tissues by using 3 different techniques: poly-A plus RNA, small RNA-seq, and iso-seq for long-reads RNA-seq. And this may help us to identify either long-range regulatory elements, promoters, or all the transcripts and transcript isoforms that are present in the different tissues. And this will be presented in the following talks. So just a summary of the open access datasets we produced. So you have the accession numbers that are for the ENA database, European Nucleotide Archive. For all the datasets, the only one that is not still available is the ChIP-seq, and it's ongoing. We are preparing the data submission currently. For all these datasets, we develop specific pipelines by using mostly the NF-core system. And we adapted some or developed some more specifically for transcript annotation. So this is the Tagada pipeline that will be presented in the next talk. One regarding the iso-seq that is also been made available on NF-core. It's a new one. And one on DNA methylation that have been developed at Wageningen University and has been used for the DNA methylation studies. Now, I will present briefly the different datasets we produced. So for the ATAC-seq, we produced 84 libraries for each species, representing the 7 tissues at the 4 developmental stages for 4 individuals each time. And you can see the quality of the datasets. So we got close to 100 millions of paired reads, which were well mapped to the genomes, with a low contamination with mitochondrial DNA, and with a median of peaks that is around 70,000 for each tissue, so that it is good. The protocol is publicly available on the Fang data portal, and for those that are interested to use the protocol we made, it has been developed by Diagenode, and you can buy the kit to do it in your lab. So this is a representation of the peak enrichment, so on chromatin accessibility at TSS for the 2 species. And you can see that for all the libraries, you have a clear enrichment of reads close to the start of the gene, so at the TSS, reflecting the high quality of these libraries. And you can also here have a view of the tracks. So this is the genes NKX 2.3 in the peaks, where you can see that the TSS is on the left. You have a nice enrichment at the day 30 of development. So at the beginning of small intestine formation, and you can see that the peak is decreasing a long time. So when you go to the newborn, it's less visible and clearly visible, and you can have different behaviors of chromatin accessibility at promoters and dynamics. But this is a nice example of something that is highly accessible at the beginning of development and is decreasing, but you can see the reverse too for other genes. This one is another example of conservation of chromatin accessibility conservation between pig and chicken. So this is at specific endodermal markers, which is named HNF4A. And you can see that the pattern of peak accessibility is mostly conserved between the two species, either as a promoter of the genes, but also in intergenic regions or at intronic or exonic regions. So this is a clear example that we can also, by looking at chromatin accessibility of this quality of data sets, identify conserved pattern of regulatory elements within a species, but also between species. So the next samples, assays, are the RNA-seq. So we made 3 types of RNA-seq, poly-A plus and small RNA-seq. So you have here the summary of the data we produced. And the main, the raw information. So we produced around 150 million of reads for the poly-A plus and 70 million of reads for the small RNA-seq, from which we map most of them for the poly-A plus RNA-seq, but less, around 30% for the small RNA-seq, but this is normal because you have a lot of duplication for the small RNA-seq. So it's a really good data set for both informations. And after that, we can look at the heat maps and hierarchical clustering for this. And, oops, sorry. And what you can see on these heat maps, so on the right, you can see a color code that is summarizing the distribution of the samples. And what you can see is that some tissues are grouping together, so like the cerebellum, the liver, and the kidney, I think. But other tissues seems to be more clustering regarding the stage. So this is the case of the upper face in the heat map. So in red, you can see that most of the endodermal tissues are grouping together at the stage 1, so during early organogenesis. And they seems to be grouped together later on by tissues for the stage 2 and stage 3. For the small RNA-seq, you can see here the distributions of the size of the reads. And you can see that most of the information is representing a microRNA, around 20 base pair of lengths. But we have also other types of RNA that should be now characterized and seems to be present either more in pigs or in chicken, and are also differently present regarding the tissues and the stages. And again, for the small RNA-seq, what we can see is that some tissues are grouping together regarding the tissue. So this is the case again for the cerebellum, the kidney, and the liver. So we can see exactly the same than the poly-A plus RNA-seq. But again, the tissues from the endodermal regions, they are grouping first by stage for the first stage of development, so the stage 1, and after by tissue when you look at later stages. Now the iso-seq, so this is another type of information that is really relevant to detect isoforms and to enrich the repertoire of transcripts we can get from short reads. You have here a summary of the information that have been obtained. And you can see that for the pig, we were able to detect 80,000, more than 80,000 genes, transcripts, excuse me, for each tissue, and 70,000 transcripts for each tissue for the chicken. The protocol is also available online. And the nice things, for example, this is an example, but it will be developed later with the talk of Sébastien Guizard. You can see that the number of genes in the pig seems to decrease during development, with more genes at the early stages rather than the later stages. But this is not always true, and you can have some example, for example, the muscle, where it's not the case. And in the chicken, it's the opposite. So we have more transcript in the later stages of development rather than the first ones. Now, DNA methylation. So we have done DNA methylation for all the samples by combining whole genome bisulfite sequencing and restriction representation of the genome after bisulfite sequencing. And what you can see is that, again, the dataset is of high quality, and it will be presented by Martin in the next talk too. The nice thing is to check the DNA methylation quality. Just an overview of this. It's to look at DNA methylation at the level of promoters. So usually we have less methylation at the level of promoters, and you can see it for the two species, either the pig or the chicken, with some differences between tissues. As you can see for the pigs, the tissues in blue, so the liver, are less methylated at early stages than later on, and less methylated than other tissues. For the chipstick, we faced some technical problems, so we are a bit late, but we have done the optimization of the protocols for all the tissues and the two species. The libraries have been produced and sequenced, and we are now doing the quality check and validation of all the libraries before being able to submit it on the ENA database. So this is just a brief overview of what we have made and what type of information you can get. So from the liver, we have most of the marks, and we have some of the marks and the stages for the kidney and the muscle, either for the chicken and the pig. So just to show you an example of the quality of the chipstick. So you can see in this a track view representing the OX complex, that you have a nice enrichment for H3K27 3-methylated in red at the OX complex that are switched off at this stage of development in the liver of fetus pig embryos. But you can see a clear enrichment at the promoter of other genes in green, dark greens, for H3K4 3-methylated, and a lower enrichment, but significant enrichments at the body of the genes around these promoters. And the last data set is the capture IC. So we have done capture IC only for two tissues, the liver and the skeletal muscles, focusing on promoters. So we made a design to capture around 15,000 promoters for each species. And the libraries have been sequenced and submitted. And this is just a view. So we are now optimizing the pipeline to do the detection of promoter and answer interactions. And it's just to give you an overview of a map of the reads at the regions that have been designed, of the promoter we designed to be captured. So you can see just the enrichment of the reads at these promoters. So finally, all the data sets we produced have been made following the FAIR principle. So all the metadata regarding samples, experiments, and analysis are publicly available on the Funk data portals. And it includes protocols, sample descriptors, experimental parameters, and analysis parameters that allow other groups to rework on it and to redo or to do other things, but with all the information that is required. All the raw sequences have been deposited in the European Nucleotide Archive, and pipelines are publicly also available on GitHub, NFCore, but some of them have also been published. So finally, we are following the FAIR principle, so our data are findable, accessible, interoperable, and reusable. And you can also easily find them through the Funk data portal. So I've wrote the internet address on the slide. And this is a capture of the screen from the Funk data portal. And you can find them. There are all the links to our raw data. To conclude, I'm on time. I'd like to really thank and acknowledge all the people that help us to do this. From the Roslin Institute, so I will mention Megan Davey, Jonathan Smith, Jacqueline Smith, Kazia Medzinska, Sebastian Guizard, Mig Watson, and Alan Archibald. From INRAE, people that help us to do the sampling, and specifically Myron Mongela, people that help us to do the analysis and experiments, Sylvain Foissac, Cyril Curillo, Servin Guillaumard, Marine Bena, Jérôme Louche, Tabata Boulage for the sequencing, Jules Saban, so Elisabetta and I to the follow-up. From Diagenode, Céline Sabatel, Laurent Servet and Vassim Lacal. From the INSERM, Sarah Djebali. From Wageningen University, special thanks to Gianni De Vos, Ole Madsen, Martin for the ChIP-seq and DNA methylation studies. And also people from EMBL, EBI, so that help us a lot for data submissions, metadata description, Alexei Sokolov and Peter Harrison. Thank you all for your attentions. We will keep the question for the end of the talks. And so I will give the floor now to Sylvain Foissac and Sarah Djebali from INRAE and INSERM. I have finished.
Hi, everyone. Can you see my screen? Can you hear me?
Yes.
All right. So thanks for joining. I'm Sylvain Foissac, and today with Sarah Djebali, we'll be presenting results obtained by the team in Toulouse, France, from INRAE and INSERM institutes about gene expression and chromatin accessibility. So thanks, Hervé, for the introduction. As Hervé mentioned, so for data production, well, sorry, I need to get this out of the way. Well, okay. So for gene expression, chromatin accessibility, data have been produced for 2 species, focusing on 3 developmental stages, each of them with 4 biological replicates, 2 males to females that are either fetuses or newborn organisms. And for each of these organisms, 7 tissues have been sampled. Okay. So in this talk, when I will use the term sample, I will refer to a combination of, for one species, of tissue, stage, and replicate. Okay, so for each species, we have 84 samples, and for each of these samples, 2 molecular extractions have been performed. So DNA and RNAs from the same samples, let's say, and DNA from the DNA, different sequencing assays have been performed, and today we'll focus on the ATAC-seq. Okay, and from RNA, actually RNA has been split into 2 different subsamples. So one part with the fraction of small RNAs that has been sequenced with the small RNA-seq sequencing protocol, and Sarah will talk about these results at the end of the talk. And the other RNAs have been sequenced in parallel with 2 sequencing protocols. So we have on the one hand the PacBio RNA-seq protocol that is generating long reads, and this is something Sébastien Guizard will present after this talk. And in parallel, long RNAs have been sequenced with the Illumina RNA-seq pipeline, the protocol that generates short reads. And this is what I will cover. So all these assays generate sequencing data. And now the question is how to get from the sequencing data, how to get to biological results. And this is the data analysis, and this is what we've been doing in this project. So data analysis for gene expression, chromatin accessibility, well, and for the other assays too, is composed like this. We have 3 parts. So the first part is the pipeline analysis. So this is something that the specificity of this part is that this is like the bioinformatics workflow that is applied for each sample. Okay, so for each combination of species, tissue, stage, replicate, we have one set of data, and depending on the data, we use a bioinformatics pipeline. So RNA-seq, we use the Tagada pipeline we designed. For ATAC-seq, we use the NF-core ATAC-seq, and smaller RNA-seq, the NF-core smaller RNA-seq. And the results is the genomic position and the molecular activity of various elements. From RNA-seq, we get messenger RNAs and long non-coding genes. From ATAC-seq, we get chromatin accessibility regions, ATAC-seq peak, we call this. And from smaller RNA-seq data, we get genomic positions and activity of microRNA genes. So for transcriptome information, the activity is gene expression values. And for ATAC-seq, the molecular activity is the accessibility of the chromatin region. Okay, so those are the elements. Now in GENE-SWitCH, we have switch. So we would like to find elements that have a significant change during development. So those are the switches, and we identify them using a differential analysis. So the differential analysis, it's performed for each species, depending on the data type. And the way it's performed is like this. For within each tissue, we look at the elements that have molecular activity element in stage 1 using the 4 replicates to get some significant, some statistical power. And we compare the stage 1 with the stage 2. And we do the same from the stage 2 to the stage 3 for each tissue. And this comparison allows us to identify, for instance, genes that are upregulated in stage 2 versus stage 1 in the given tissue, or downregulated, and the same bit from stage 2 to 3. So those are the switches, and this is what the differential analysis gives us. So why do we do that per tissue within each tissue? It's because, well, tissue is often like one of the most driving factor in terms of biological differentiation in gene expression or chromatin accessibility. So we have much better results by focusing on each tissue separately. Of course, then once we have switches, we can compare them between tissues. And this is something we've done. So this is completed. And the last part of the analysis is the integrative analysis, where the goal is to integrate all of the data from gene expression, chromatin accessibility, and the other assays, methylation, ChIP-seq, and so on, into an integrative analysis. So this is still in progress due to some delays in getting some of the data, but we've already performed some like cross-check and between expression, ATAC-seq, and methylation and expression, for instance, and so on. So this is ongoing. Okay, so now let's dive into these transcriptome data. So to analyze this transcriptome, the first part, the pipeline analysis part, the challenges were the following. We needed something that was reliable, reproducible, scalable for the the fair principles and stuff. And a specific challenge for that was to integrate PacBio long reads and Illumina short reads. As I said, transcriptome was sequenced with both technologies. So it was important for us to get the best of these two technologies. So, well, RNA-seq data pipeline are quite frequent, and you can find a lot of them to, if you give an annotation, some transcriptome data, you will get gene expression values for your genes from the annotation. So that's not a real issue. And with the NF-core RNA-seq pipeline, the one that by default is the one that is available by default, we have these factors: reliability, reproducibility, scalability, that are quite covered. The thing is, it doesn't generate one integrative annotation, and so far, as we know, there is no pipeline that is available to mix and to integrate these two technologies there. So we had to design our own pipeline for GENE-SWitCH, and this is the Tagada pipeline. It's a pipeline for transcript and gene assembly deconvolution analysis. So it's designed, well, just as an overall picture, it's designed in such a way that you can provide it a reference annotation, some transcriptome data, and then it will integrate them, okay? And not just quantify the gene expression for your reference annotation, but also improve the existing annotation to provide a new one and quantify transcripts and genes from the new annotation. It does all the way, it does everything. So, and it's very convenient for us in the context of GENE-SWitCH, because we could provide the PacBio long reads annotation derived from the Sébastien Guizard pipeline. He will present later. We could provide that as a reference annotation and the short reads from Illumina sequencing, so that it could, merge both information. And this is a pipeline that we published recently this year. Well, actually, I realize it's published in December, so it's in the future. So it's a very advanced technology from the future. Okay, so that's the sequencing, RNA-seq sequencing pipeline, and we get genes from there. Okay, so we get a lot of genes. So, sorry. I was distracted by the chat. I shouldn't look before. So we get 30,000 genes. So this is number of genes from the Ensemble assembly version, and the Tagada is on the right. So we have like 1 to 1.3 times more genes than in Ensemble, and 3 to 4 times more transcripts. So this is something expected, as Ensemble is delivering a reference annotation for the community that is built on all the transcriptome data that was provided worldwide. So here with GENE-SWitCH, we can focus on because Ensemble annotation has to be quite is a consensus and has to be a bit conservative. Here we can afford to get much more resolution in the transcriptome landscape, and to get a lot of long non-coding RNAs, and this is why we expand this catalog. Tagada genes, so a lot of them are non-coding, with, of course, much lower expression levels. A lot of them, of these genes we find, refer to existing genes that are already known, but we have a lot of new ones. And of course, we've run a lot of quality controls for these transcripts, with the coverage of ataxic peaks in the promoter region, the transcription start site, polyadenylation site at the end of the genes or the transcripts, and well, canonicity of the splice sites for the introns. So this is all published in the paper from the future there. Okay, so that's about the genes and transcripts. Now, can we find switches? And we did find switches performing these differential analyses between stages, consecutive stages, stage 1 and 2, 2 and 3. So those are the numbers. Those are very high numbers. Objectively, this is very high. This is not what we expected. Using these 4 replicates, so it's not a lot, but it's enough to get statistical power. And one gene out of 3 genes or 4 are detected as potential switch in different tissues. This highlights the fact that development is quite a strong biological process with drastic changes in transcriptome. Of course, here, this is the number with no filtering applied. This is just statistical changes. And of course, depending on the fold change we look at, we can focus on more stringent results. What's interesting then is to ask if, yes, genes that are identified in one tissue are the same in the other ones. And this is the study of tissue specificity. Tissue specificity. Specificity of the tissue for the genes. So are they genes that are common to the different tissues here? And this is what we have in the next slide. So, of course, so this number of tissues where you can see a gene. So we have 16,000 genes, for instance, here in chicken, from stage 1 to stage 2 that are specific to one tissue, and the other one are shared by 2, 3, 4, and so on. Okay, so tissue-specific genes are not the majority, because if you add up all the other values, you get something a bit higher, but it's a most frequent category. And we have a very low proportion of genes that are common to 7 to 8 tissues and that are identified as switches, okay, in a lot of tissues. So those are genes we will focus on a bit later for functional analysis. It's interesting results. Okay, so as I said, we have Tagada is not only providing results at the gene level, but also at the transcript level. And this is very interesting because, sorry, I forgot a bit before. Once we have these genes, of course, we look at the functions of these genes and we can characterize functional analysis. So whether we have enriched gene ontology terms in the different population of genes we identified. And as expected, we have functions that are quite consistent with the tissue of origin. For instance, for skin, we have hair and skin development functions, which is quite expected, of course. Muscle, I guess, like energetic and mitochondrial processes. And from stage 2 to stage 3, in chicken, for instance, we have functions that are significantly enriched in the genes we find as switches, and that are synaptic and nervous system development and so on. So it's all consistent in terms of biology. So back to the transcripts. Since we have expression data for an annotation for the transcripts, we can investigate the landscape of the transcriptome in a very high resolution manner. So this is an investigation on alternative splicing. So alternative splicing, what it is, it's the fact that from the same gene you have transcription, and then you can have splicing that is different transcripts. So we call an alternative splicing event. Alternative splicing event is a local difference in the exon-intron structure between transcripts. Okay, so how do we find that? For instance, the most frequent event is exon skipping, which is quite common. And we find it, this corresponds to this. If, for instance, in the genome at the DNA level, you have a gene X with 3 exons here, A, B, and C. And between the exons, you have introns that are spliced during the splicing process to generate transcriptome with, for instance, transcript X1 that is the results of a splicing from exon A to C directly, and transcript X2 that is that results from the integration and the inclusion of the exon B. So whether the exon B is integrated in the final transcript, we have this local variation that is observed, and this is what is called exon skipping. So to detect this, we perform the analysis in 2 steps. First, we use the expression level from the transcript to filter transcripts within each sample. We identify the list of transcripts that are expressed. We find a minimum expression threshold for that, very basic, so that we retain all the expressed transcript. And then we use a specific tool that's called AstaVista that can compare the exon intron structure of all of the transcripts in a pairwise fashion to identify all the variations like the exon skipping event. And all of these events are listed by AstaVista. So if, for instance, these 2 transcripts are considered as expressed in one sample, then AstaVista will report 1 exon skipping event here. But if another gene further in the genome, gene Y, has 2 transcripts with a similar structural variation, although it's not the same position. Exons can be longer. The sequence obviously is different, but if it's the same variation, structural variation between 2 different transcripts, it will list another exon skipping event. Okay, and this is done for all of the variations that you can find between 2 transcripts. So we have like the all-stars events that are the usual suspect ones. They are listed everywhere with alternative donor site, alternative acceptor site, a variation of the 5' end of an exon, intron retention, if in one transcript part is included or not, with both transcripts being expressed. I insist on this feature. Well, all these events are counted. Okay, so within each sample, we get a list of events, and we just retain the number of these different events. So here, those are the ones that are the most frequent ones, but we have hundreds of possible variation between 2 transcripts. So then we can just plot these values, and this is what we observe. So for instance, this is the relative proportion of these events I mentioned with different shapes, and colors are representing the developmental stage. So in chicken, the most frequent event, as expected, is exon skipping. Then we have intron retention, then alternative acceptor and donors. Those are double events, not very frequent, and then all of the others are represented by these crosses here. So exon skipping is the most frequent event, as we expect, except that in one tissue, we find more intron retention than exon skipping. This is a swap that is very specific to this tissue, and it's liver. In chicken liver, we have this interesting change here. Otherwise, the profile we have is that generally exon skipping and intron retention are quite exhibiting a symmetrical profile. In pig, we have also exon skipping, that's the most frequent one, and intron retention. What could be interesting is that in pig, here we have a very high frequency of exon skipping and a low frequency of intron retention in the three cerebellum samples, so the brain. And there's another outlier that is identified here with a huge difference between exon skipping and intron retention. That's specifically the late stage of muscle development, so newborn, which is also quite interesting in terms of biological effects. So we're not expecting, because all of the other events are quite stable in frequency, so we're not expecting a lot of variations there. So this prompted us well, it raises the question of how far can we get in terms of resolutive power of this analysis based just on alternative splicing events. I just want to make clear that here we are not using the expression value to perform the analysis, just to filter the transcripts that we are considering. So basically, when we do a transcript analysis, we are using all the expression values, and it's a quantitative metric, right? It's expression values go from 0 to whatever. And here we are reducing this value to a binary information, either 0 not expressed and 1 expressed. And we are comparing the transcript based on this value. So it's a quite drastic reduction of information. And then we count the number of events. We do not keep the genes IDs of this event, right? We have a number of exon skipping events. They can be anywhere on the genome. We just retain this number of intron retention and so on. So each sample has these numbers there, the frequencies, and we can plot these. We can use these, for instance, dimensionality reduction analysis, PCA, to look at how samples distribute using just these events. alternative splicing features. And this is what we get with on these 2 dimensions here. We have samples, for instance, that cluster according to tissues and stages. For instance, on the left, you have muscle replicates on top. Those are from stage 1. Then we have stage 2 samples, and then stage 3. On the right, we have liver samples. So this is for chicken. And for pig, we also have some clustering there with the liver, some stage 1 that cluster there, the stage 2 here, stage 3. So it's quite remarkable that with such a contraction, such a reduced information level, we are able to discriminate and to find some biological information in these data. So it's a well, quite original and interesting results we get. All right. So now I will let Sarah jump in and
yes,
follow this presentation.
thank you. Can you hear me well?
Yes.
Okay. So yes, good afternoon, everyone. So I'm going to continue with the analysis of the small RNA-seq data and ATAC-seq data. So for the small RNA-seq data, we use the small RNA-seq pipeline from NF-core, where we gave the MIR-based database as well as our short reads in 84 samples. And we obtained the expression of the MIR-based microRNAs in the 84 samples, as well as a new small gene annotation for GENE-SWitCH that was made out of our 84 experiments with the MIR-DIP2 tool, as well as bed tools, and some filtering on the number of experiments where we want to see microRNA. So in MIR-base, we have about 1,200 microRNAs in chicken and about 500 in pig. And so with the NF-core pipeline and the bed tools, we managed to obtain about 2,200 small RNAs in chicken and 1,600 small RNAs in pig. And we see that in both species, we have about 1/4 of these small RNAs that are already known in any species, and about 3/4 that are novel. The next slide, thank you. Then we did, so I think Hervé showed you something like this already, but here I show you for the 2 species the hierarchical clustering of the 84 samples based on the MIR-based microRNA expression. So you have a heat map where the color represents this correlation between expression of these microRNAs in a pair of samples, and where you have the colors that represent the different attributes of the experiments, developmental stage, tissue, and sex. And here we see a picture that is quite similar to what we get with the long RNA-seq, which is that you have some tissues where all the experiments of the tissue cluster together independently of the, all the samples from this tissue cluster really together. This is the case for cerebellum, liver, and kidney. And you have other samples where the first clustering is by stage. All the stage 1 samples cluster together. And we see that for both species and quite similarly to long RNA-seq. Next, thank you. So now we also looked at the number of experiments where we see microRNA detected. So I have to first to say that when I say we obtain about 2,000 microRNAs in our novel annotation, it's actually we filtered, we only kept the microRNAs that were seen in at least 2 experiments. And here what we see in the known microRNAs are in red and the novel are in green. And of course, we see that the known microRNAs are detected in more experiments than the novel ones. But still, we can see that some novel ones can be seen in many, many different experiments as well, like more than 60, for example. And we need to look into those in more details. And yes, so now I'm going to talk about the chicken and pig chromatin accessibility landscape during development, thanks to ATAC-seq data. So for ATAC-seq, we use the NF-core ATAC-seq pipeline, giving it as input the ensemble gene annotation and our 84 experiments. And doing so, and this provided us a list of consensus ATAC-seq peaks, which are chromatin accessible regions across all our experiments. Apart from, so we have this list of peaks, and we have also the quantification of their accessibility in the 84 samples. So doing so, we get about 350,000 ATAC-seq peaks per species. And quite expectedly, the majority of those peaks are intronic. 45% of them, then 20% are intergenic, and about 10% overlap ensemble TSS. Yes, so now we looked at the distribution of the ATAC-seq reads, mapped reads around genes. And so what we see is that for the vast majority of the samples, we have a clear peak at the TSS of the genes, and a depletion at in the gene itself, and then a little bump at the TS. Some samples, we see exactly the same picture for pig. We have to say that some samples have quite flat distribution, and actually there are very few of them, but they will be removed because something went wrong with them. Then yes, we also did the same analysis as for long RNA-seq. This to say the characterization of the switches, the accessible region that switch between two consecutive stages. And again, like for long RNA-seq, we see that the vast majority of the switches are tissue specific, and very few are seen in all tissues. Maybe this is to a less extent than for the long RNA-seq, as I can see, yes. But still, yes. And here it's another way to represent this information, where you have two circles, and the inner circle that is represents actually the number of switches that are tissue specific, and we see that the majority of the switches are tissue specific. We have some problem with the skin in pig, and this is also those are some samples that we will have to discard. But apart from that, we see similar trends between the two species, and quite nice data otherwise. So as a conclusion, so for the three types of data that we presented, we have already, of course, we have chosen the pipeline, and we have either developed or deployed the pipeline for these three kinds of data. We also have performed a differential analysis between stages, and now we are in the process of integrative analysis with some steps that have already been performed, like for example, the long RNA-seq data has been compared to the methylation at Wageningen University, also with the ISO-seq at University of Edinburgh. We have crossed also this information with small RNA-seq and ATAC-seq. So we are in the process of performing all this very interesting integrative analysis.
Yeah, and sorry, Sarah, for the alignment.
No problem. It's okay. And so as a perspective, we want to compare the switches that we find across assays. That means the switches we find for different kind of functional elements. For example, we are interested in knowing that whether if we have a gene that is that is switching between stage 1 and 2. Do we have a differential accessible region at its TSS that is also switching between stage 1 and 2 or not? And we want to, of course, integrate data to derive the regulatory relationships and their switches across stages. And we also want to use comparative genomics in order to get a better understanding of the evolution of these regulatory relationships in vertebrates. And this will be performed thanks to our active working group from INRAE in CERN with Servin, myself, and Sylvain, but also at Wageningen University with Martin and Ole Madsen, and at University of Edinburgh with Sébastien Guizard. And so, yes, we would like to acknowledge, of course, all the WP2 participants, and in particular, Servin at INRAE, and Cyril Curillo also at INRAE, Yoni DeVos for the methylation at Wageningen University, Martin also at Wageningen for methylation and ChIP-seq, as well as Ole Madsen. And then Sébastien Guizard at University of Edinburgh for ISO-seq, and Alain Archibald for coordination. WP1 with Hervé Atloc from INRAE, and for the coordination, Elisabetta Giuffra and Hervé Atloc. And thanks for your attention. I'll be done, sorry.
Maybe we have some time for questions if there are questions. I think we had something in the chat, right? Yes, so is there interest? So I'm reading a question from the chat. why Steve the mic? Is there interest in running clustering analysis across stages in addition to pairwise and differential expression analysis? Yeah, so maybe it was before I mentioned this. Yes, it was a question in anticipation. Yeah, so we did both actually, as usual. We did different kind of differential analysis. So the one I mentioned was within each tissue, right? Well, I want to make sure I got the question. Otherwise, Chris, just correct me. Clustering analysis across stages. Yeah. So interest clustering analysis. Yes, so clustering analysis would be like the something we Sarah presented with hierarchical clustering, right? So something like this. And well, we did that. We did that for long RNA-seq data as well. And we did that for chromatin accessibility. And as Sarah mentioned, yes, we observed a bit the same pattern with samples that cluster according to tissues, or samples that cluster according to stage, often from stage 1. So this for some of the tissues.
Ben, can you hear me?
Yes.
What I was meaning was not clustering by sample, but clustering by gene. So a gene co-expression analysis to look at regulatory groups of genes across time, because of course you're doing the ATAC-seq data and looking at regulations. So I was wondering if you were thinking about regulatory,
Yes.
co-regulation, that kind of thing.
Co-regulation, exactly. So this regulative analysis, something we've done on other data as a preliminary exploratory research. It's basically a gene network, right? Analysis. Yes, that's it. So this is something we are looking into. Yes, we are planning to do that. So there are different ways to do it. Either assay by assay, for instance, expression network, by using correlation values between samples for all of the genes, correlation values to build the network. This is something that's interesting. It works quite well. Usually, you really get clusters within the network. You get clusters that are based depending on how many clusters you want to identify. You get clusters that basically correspond to tissues, but we would like to expand this to have more resolutions in the results. We would like to do is to perform this kind of multi-dimensional network analysis by combining different assays. So this is a project we have with Sarah and Servin, based on the combination of, like for instance, promoter accessibility, gene expression, then also link microRNA expression to the long RNAs that can be target, from the microRNAs using target prediction, and combining all of this with like the capture IC that helps bring some chromatin accessibility marks that are outside of the genes to their potential target genes, and getting this kind of large network analysis, we would get to the regulatory network. And this is like the ultimate goal of this analysis step.
Thank you.
Yeah, thanks to you. You have time for more questions if anyone wants to jump in.
We can move on to keep on time, and we will have a question and answer session just after the coffee break.
Thanks.
Yeah. So Martin.
Thank you very much. Okay, thanks for inviting me. So today I will give an overview about some of the work on the methylation that we've done at Wageningen University. So this is actually mostly Jani de Vos, her work, and she will defend her PhD thesis next month. We also did some preliminary analysis on the integrative work, so trying to combine ATAC-seq, ChIP-seq expression to construct chromatin states. So I'll talk a bit about that tomorrow when we talk about the integrative work. So also for the methylation, we have, of course, a large data set. So for the RBS, which is kind of a reduced representation of bisulfite sequencing, which mainly targets, I think, promoter regions. We have in total 63 samples, so we have the same, I don't think I need to introduce the tissues, so we have the same tissues and time points. So that's both for pig and chicken. So when we looked at the methylome, the first thing that we saw is, it's probably not working. Okay, I can't use a pointer, but I'll try to explain. So the first thing that we saw was that the methylation levels for the liver, so for the liver day 30 and the liver day 70, they were significantly lower compared to all the other samples. And this was very clear. So I think overall it had a 20% less methylation level in these two liver stages compared to any of the other samples. And also if we looked at the clustering of the RBS data, we saw that also there that it's probably not visible because I also can't point, but also there we saw that a lot of the stages on day 30 actually clustered together at that stage. So that probably means that the tissues were not differentiated enough to really form a cluster together with its tissue type. So one other thing that we saw, so in the middle, you see the two liver stages. So on the bottom of the slide, so you see liver day 30 and liver day 70. And what we saw is that for these two liver stages, we hardly saw any fully methylated sites. So that means that probably when we did the methylation analysis on liver, that there were cells, I believe, that were probably not methylated at all. And then, of course, it's when you combine that, you'll see that you have very little fully methylated sites because you don't see that peak on the, let's say, on the tail showing the fully methylated sites. So probably it was kind of a mix of cell types with different methylation patterns in those two liver stages. For the chicken, we did not see this for liver. So for the chicken, we saw relatively even methylation levels across tissue. When we looked at the clustering, we saw that in chicken at day 8, so at the first stage, we did see that many of them already clustered together by tissue type. So that means that probably in chicken, by that stage, the tissue has already differentiated enough to cluster by its tissue type. So this was also different from what we observed in pig. When you combine the methylation data with the expression data, you see that, especially of course at the transcription start site, so that's the just upstream of the gene, you see a clear drop in methylation, especially for the genes that have the highest expression. So in this case, the purple line. And you also see that in the gene body, generally, you again have quite high methylation and then dropping again a bit at the transcription termination site. What we saw in pig is that for the genes that had the highest expression, we saw that also after the gene body, we saw a clear drop in methylation. And this we did not see for the chicken. But it did show us that at least the correlation that we expected between methylation and expression was what we expected. Next, I also looked a little bit into the methylation patterns of repetitive elements. So in general, repetitive elements are quite highly methylated. This is also expected because many of these repeats are silenced. So they should not be active or they are not functioning anymore. So generally that means that they're just methylated highly. But when we look at, there was one specific type of repeat, so this ERVK, which actually had a relatively low methylation level. And it could be that this type of repeat is a bit younger, and therefore there might be still some repeats active. And therefore we see this lower methylation pattern in this specific type of repeat. Also, when we look at, these are two types of repeats. So in pigs, so this is ERV1 and an L1 repeat. So these repeats are relatively common in the pig genome. And here what we also saw is that also for the liver, those two liver stages, we see a very clear lower methylation level in these two repeats. What was also interesting, if you look at the ERV1, but also at the L1, you see that the methylation pattern actually within the body of the repeat, it drops compared to the neighboring regions. So yeah, this was kind of surprising because I would expect that it would be also highly methylated across the repeat body. In chicken, this we saw actually a different picture. So in chicken, we saw on top, you see the CR1. We saw that the methylation levels within the repeat body actually increase. And this is also, I think, what we would have expected. What we also saw is that for lung at hatch, we saw significantly lower methylation levels at these type of repeats, whereas for brain at E8, so at the first stage, we saw higher methylation levels at these type of repeats. So this was really kind of an interesting also distinction between the chicken and the pig that we saw on the repeat level. Even though we did not have the ChIP-seq data yet, we did try to see, okay, how are the methylation levels in different chromatin states? So to do that, we used chromatin states that were predicted by the Panadol paper 2 years ago. And they defined different chromatin states. So on the right, you see all the chromatin states that they defined. And we looked at, okay, what is the methylation level in these different chromatin states? Of course, we want to repeat this now with the ChIP-seq data that we have from our own samples. And what you see, and this is also expected, the active transcription start site, we see a relatively low methylation level compared to the neighboring region, so you see a clear drop. Also transcribed, a gene has a relatively low methylation level. And also if we look at enhancers, so this was actually quite interesting, if we look at active or medium active enhancers, we only see a very slight drop in methylation levels. And if we look at weak enhancers or poised enhancers, they actually have higher methylation levels compared to its neighboring region. So this is a very clear distinction between these enhancer types in their methylation pattern. For chicken, we kind of saw the same picture. The only difference that we saw in chicken is that the enhancer states actually have a bit higher methylation pattern, and you also see a slight increase in methylation pattern for the chicken compared to the for the active and medium active enhancers. So this was again a slight difference between the pig and the chicken, but overall, if we compare these chromatin states and if we look at the methylation level at these chromatin states, we saw quite similar picture between pig and chicken. So Yanni also did some analysis on checking the transition between different methylation patterns across different time points. And what she saw is actually that there are quite some regions that transition from lowly methylated regions to fully methylated regions, or the other way around. So generally, lowly methylated regions are considered to be active enhancer sites, whereas fully methylated regions are, well, they're usually indicating repression, so they're probably not no activation. And we saw very little change comparing the unmethylated regions. So the unmethylated regions are usually the active promoters. So then you really have no or very little methylation. But we did not see a lot of changes from unmethylated to lowly methylated or fully methylated. And that was actually the same for pig, also when we compare that in chicken. And Yanni has actually, in her thesis, she has two papers that will probably be submitted soon, and then also more of this will become available to the scientific community. If we look at differential methylation analysis in pig, the large blue bar is again the maturation phase for the liver. So there you see you go from liver day 70 to newborn. And what we've seen before, you see a large change in methylation patterns. So that's why if we look at differential methylated regions, then this liver maturation phase is clearly an outlier and shows, and we see very clear differences in methylation, also when we look at differentially methylated sites. We also checked whether if we see these differentially methylated regions, how that affects gene expression. And we saw that most of the, so this is an example in brain, and we saw that indeed, if we see that a certain region becomes hypomethylated, so it means it gets a lower methylation, then generally that leads to an increased expression of the gene. However, we did also see some exceptions, so where we actually saw that even though the methylation goes down, we also see a downregulation in gene expression, which is of course something that generally we don't see. One thing that we also did is we looked at how many differentially methylated regions and sites we have from the whole genome bisulfite sequencing and from the RBS. And in this case, we saw that especially at the developmental stage in chicken, in brain, we saw many differentially methylated sites. And also if we looked at the function of these genes, often they correspond to the tissue type. So that was also a confirmation that the analysis that we do on differential methylation points to genes that are important for tissue development. So with that, I would like to conclude. So we did an in-depth analysis of the methylome across development and tissues. We specifically looked at repetitive elements where we, I think, made some interesting observations. We also looked at methylation patterns across epigenomic states. And tomorrow I will show some preliminary results where we try to build epigenomic states from our own ChIP-seq data. And we looked at differential methylation across tissues and time points. One thing that was very prominent is that those two liver stages, so the fetal stages in liver, show a much lower methylation level compared to the other samples. And we saw specifically that they lack fully methylated sites. So probably there were cells that were not methylated or had a very low methylation in combination with cells that had a higher methylation. And this is something we have to dig deeper into to see what's going on there. Thank you.
We have time for 1 or 2 questions.
You mentioned differences in methylation, specifically in liver compared to the other tissues, lack of heavily methylated genes. Can you correlate the lack of methylation with the liver expression? Yeah, so that's a good question. We did that, and we did not see a very prominent difference in gene expression. So that was also quite surprising. So even though we see this very distinct methylation profile in those two liver stages, it was not reflected in the gene expression, if I'm correct. So, yeah.
Martin, can I ask a follow-up then?
Yes.
Is it possibly a technical issue with those particular samples? Could they have not gotten the same level of I mean, I know you did whole genome bisulfite, but then also did reduced representation. I'm just wondering if you don't get as much bisulfite treatment, would you get a difference in the output?
Yeah, so again, thanks for the question. So as far as I'm aware, we see the same pattern also in the RBS samples. So I don't think I would not expect it's contamination.
Okay.
But yeah, of course, we are not 100% sure, but I think because we see it both in the whole genome and RBS, I would suggest that it's correct.
Can I add to this? Can you hear me?
Yeah.
Yes.
Because from the whole genome bisulfite sequencing, we also got let's say the positive control of the spikes in the data, and they all look the same. So I would suppose that the treatment of the bisulfite have gone well for these samples as well.
Do you know if that's seen in human fetal liver? I think the data is out there. I'm not sure, but
It is seen in mice, as far as I'm aware.
cool.
So there is probably something in liver that are special in mammals.
But maybe not related to gene expression.
No.
Just to mention, so the liver during the late pregnancy state changes completely of destination. First, it is an hematopoietic organ, and then before birth, it's this hematopoietic function is lost. And I was wondering if this evidence, these indications could recall to that biologically somehow, and also if other data sets are showing something quite special in the switch between 70 days fetus and the newborn.
Yeah, so I'm aware of this change, let's say in liver at a certain point. I think techniques like single cell at some point might help to disentangle what's going on exactly. But for now, I think, I think that's probably the way to go. Yeah.
So maybe we should think of some the convolution approaches also to our data. If we could use what is already available, like a single cell atlas in pigs and so on.
Yep.
Maybe to also try to answer these kind of questions.
Yep. But one of the unique features, of course, of this is that we have these developmental stages, and also many of the data sets that are available are, of course, from mature or newborn stage.
Yeah, so not, yeah, correct. Yeah, not suited completely.
So we are just finishing the session now. So we have the coffee break for 20 minutes, if I remember well. And after we have the presentation of Sébastien Guizard on the ISO-seq results on a video. Yeah, it's a video. And after that, we can follow with some question and answers on the different presentations. So see you in 15 minutes. Thank you very much for all the speakers and the audience.
Does that work? Can you hear me?
Yes, I can hear you.
Okay, and you can see the.
Sorry for the misunderstanding. We just have a video before you.
Yeah.
But when the video is finished, you can do exactly what you do.
Yeah. Okay. Sounds
Perfect.
good.
You're
Yeah,
awesome.
thanks.
Thanks.
Yes. So, hi everyone again. So we will have now a video from Sébastien Guizard. There is an echo. So the next talk is a video from Sébastien Guizard on the ISO-seq results we obtained from pig and chicken using the different tissues of the GENE-SWitCH project.
Hi, my name is Sébastien Guizard, and I'm very sorry for not being here today for presenting you my work which is uncovering the early transcript landscape with ISO-seq. And first, I'm going to talk about my personal objective, which were to produce a gene annotation and transcript or isoform annotation with a technology called ISO-seq. And for doing that, I had to develop a pipeline to make the analysis automatic and also to make it reproducible. One question you can have is why using ISO-seq when we are already doing an RNA-seq annotation? So we can compare those two technologies. RNA-seq and ISO-seq have different advantages and disadvantages. First, RNA-seq, it's a short-read technology, so it can sequence up to 300 base pair with an accuracy of 99.9%. The quantity of data that is produced in the same run is very high, so we have a very high depth of sequencing, and for a very cheap price. But the annotation requires heavy computation. For ISO-seq, it's a long-read technology, so it can sequence up to 20 kbs with a lower accuracy of only 90%. But with some tricks, it's possible to increase it to the level of Illumina. For the sequencing depth, it's lower than Illumina, and the price is more expensive. However, the quantity of computation needed for the annotation is much more lower, and the main advantages of ISO-seq, it's its capacity to detect the exon chains in genes, and so make some isoform detection. So it's a big win for ISO-seq. So how does ISO-seq work? So first, you start by extracting your mRNA, then you transform this mRNA into double-stranded circular DNA, and this molecule is then loaded into the sequencer that will produce a very long molecule like this that contains several copies here of the original mRNA. And as you can see, those copies have errors inside, which is explained by the biochemistry. So before using this for the annotation, you need to do a post-treatment. So you started for each subread to generate a consensus sequence from each copies. So here you have the consensus sequence, and this is done for each subread. So you will have a series of circular consensus sequence that still contains the primer sequence here and also the poly-A tail. And so the first step is to remove those primer from those sequence, then to remove the poly-A tail from the sequence. And in the end, you have what they call the full-length non-chimeric reads. Then you take those sequences that you map on the genome here. They have a line on different regions of the genome. They stack up together. And the last step is to collapse those alignments to produce clean gene annotation and transcript annotation. So for doing that, I have developed a pipeline called NF-Core ISO-seq. So very quickly, NF-Core is a bioinformatics community that promote high quality pipeline and also high reproducible analysis. And they gather all this pipeline on their website and on a repository on GitHub. As you can see here, my pipeline is on NF-Core ISO-seq and have 70 stars. Yay! And also I published this pipeline in a paper in bioinformatics. So how does it work? So here is my pipeline. So as you can see, it's composed of 3 main parts. So the first one is a subread processing that generates the CCS and cleans them to obtain the FLNCs. Next, those FLNCs are loaded into the mapper. So the user have 2 choices for the mapping. So they can use Minimap 2 or Ultra for doing that. The alignment produced by the mapper are then sent to the last part, which is the collapsing of the alignment with Tama. And the last one is the merging of all annotation, because from the beginning, the dataset have been split into small batches to be treated in parallel, so the whole analysis is very fast. So now the most interesting part, what result do we have with IsoSeq? So as a reminder, for the dataset, I had three development stages for 7 tissue for the 2 species, the chicken and the pig. And the important information here is that the library have been selected on the presence of a cap and also a polyethyl. So that means that we have analyzed the complete mRNA sequence. So the first information that we have a look is the number of reads that we have produced with those IsoSeq. And here you have the result for the chicken. And you can see we have generated more than 66 millions of reads. And over these 66 millions, only 256,000 of them didn't map on the genome. So we are very happy with this number. That means that our sequencing is high quality. For the pig, it's very similar. We have generated 63 millions of reads, and even less reads didn't map on the genome. So we are also very happy with that. Before talking about the annotation, I had to explain you I have made a filter on it, because at the output of the pipeline there is still bad annotation in it. And so I decided to remove all annotations that are not supported by minimum 3 reads. Also removing all artifacts that are generated from RT switching and also from non-mediated decay RNAs. I have done that using Scantifree. And I have also removed all artifacts from internal priming. So what is it? It's artifacts that are generated due to the usage of primers that contain poly-A inside of it. Because of this poly-A, the primer can bind to an internal poly-A, for example here, and this binding will create an annotation, a false annotation here from IsoSeq. And so to discard those bad annotation, we just have a look to the bases after the last exon, and if there is too much A there, so we just discard this annotation. So how many genes do we have found with IsoSeq? So here you can see the result for the chicken, here for the pig, and on the top row you have the number of genes that have been annotated with IsoSeq, and on the bottom row you can see the number of genes that are annotated in Ensemble 108. And as you can see, we annotated 22,000 and 25,000 of genes, but we can go deeper in the numbers by splitting the numbers between the new annotated genes and genes that have been already annotated. And as you can see, for the chicken, we have found 7,000 new genes, and for the pig, we have found 10,000 new genes. So we are very happy with that. But what about this transcript? So for the transcript, as you can see, with the same layout, we have annotated twice the number of transcripts compared to Ensemble. So it's a huge win here. But if we go also deeper in the analysis and split by new discovered transcript, you can see we found only 11,000 new transcript and 14,000 new transcript for the pig, which is very low compared to the number of genes that we have detected with IsoSeq. Here, 7,000 for the chicken and 10,000 for the pig. But these numbers are explained by the next figure. Here, as you can see, it's a number of transcripts per genes. And as you noticed, the new IsoSeq annotation mainly have only one transcript per gene, which explains this ratio between the two gene and transcript. For the transcripts that have been already annotated by Ensemble, you can see we have quite a similar pattern to Ensemble, at the exception that we have enriched the genes with a new isoform. So the beauty of this pipeline is that it uses TAMA, and thanks to TAMA, we are able to split the data per development stages or tissue or even the time point, and so we can have a look to the landscape of the transcription during these different features. So here you have the number of gene and transcripts that we have found for or at the level of the development stages. And as you can see, for the chicken, there is an increase during time of the number of genes that are expressed during the development. And this is the same for the transcript. But if you have a look to the pig, we have the reverse. We have a decrease of the number of genes and transcripts that are expressed during time, at the exception of these two stages where the number are pretty the same. So we can have a look to the same thing, but at the level of tissue. So here on the tissue, you can see we have some top tissues, which are the ileum and the lung for the chicken. But however, we don't see any separation in the tissue, not like the pig, as you can see, where we have a very one top tissue, which is the lung, which have the most gene and transcript expression. After, you can see a second group of tissue with ileum, muscle, skin, and kidney, which are mid-express as well for the transcript. And then you have a third category where you have the brain and the liver, which are the less expressed tissue. And now what about the time point? So as you can see here, for the result for the chicken, on the left you have the genes and here the transcript. So as you can see, there is different pattern of expression during time for each tissue. For example, the brain and the lung and the skin have an increase and then a decrease of the gene expression. The ileum and the kidney have a constant increase of the gene expression. The liver have a decrease and then an increase. And to finish, the muscle have a constant decrease of the gene expression. And as you can see, the transcript follows the same pattern of expression. But the interesting thing is when you switch to the pig, you can see the tissue have different pattern from the chicken, but also between the genes and the transcript, there is also different pattern of the gene expression, which means that for some tissues there is a change in the number of transcripts that are number of isoforms that are expressed, and so we have some isoform switching for this result. Another interesting thing that we can have a look with this result is how many specific genes that are expressed and how many specific transcripts that are expressed for each depth stage, tissue, and time point. Here you have the number of specific genes and transcripts that have been detected for each development stages. And for the chicken, you could notice that the stage with the most specific genes and transcripts are when they hatched. For the pig, it's a little bit different. In terms of genes, it's for the stage 30 dpf where we have most gene. But for the transcript, it's at the newborn stage where we have the highest number of transcript. On the level of the tissue, we have the same pattern as before, where with the lung, which is the top tissue for the specific genes and tissue. For the pig, it's also the lung as the second place, but it's more the brain and also the lung for the transcript. And at the level of the time point, for the chicken, you can see that the top tissue and stage is the lung at the stage hatched, and this is the same for the transcript. For the chicken, for the pig, you can see that the top tissue are the brain at the stage newborn, the lung at the stage 30 dpf, and also the muscle at the stage 70 dpf. And it follows the same pattern for the transcript. So earlier I told you that IsoSeq was better than Illumina, but it's not really true because each technology has their own advantages and disadvantages, and the best that we can do is to merge the annotation, the RNA-seq annotation and the IsoSeq annotation to make a new super annotation called TagIso. So it has been done with the help of Sarah, Servin and Sylvain from Zinae of Toulouse, and we have generated two TagIso annotation for the chicken and the pig with a higher number of genes inside of it, also a higher number of transcripts for each of them. And the very interesting thing is you could believe that it's the PacBio annotation that will improve the Illumina annotation, but it's not always true, because we have some isoseq annotation that have been improved by Illumina annotation and on the both sides of the annotation. So it was a very interesting result. So to end my presentation, I would like to thank all those people again for all the help they give me during these 2.5 years, especially Mick Watson that hired me, Jacqueline and Alan that support me during all this time, and also a special thanks to those 3 guys that helped me a lot and are very nice, and I was very happy to work with them. So thank you for listening to me. If you have any question, do not hesitate to come to see me tomorrow and ask them. So have a good day.
Now we have the question and answer sessions. So from the chat, we saw some. One is, what was the correlation between data generated by RRBS and WGBS for the samples that were analyzed by both methods? It was, I think, a question for Martin or we have a microphone.
Okay, so the question was the correlation between whole genome and RBS?
Yeah, between samples.
Yeah, so we checked that and the correlation was quite high. I think 0.8 or something like that. But I don't know the numbers by heart, but I think the correlation is relatively high, yeah.
Another question, thank you, Martin. Another question for Sarah and Sylvain. Why do we have less microRNAs in pig compared to chicken?
So in MIR-based, actually the microRNAs are much better annotated than the pig ones. Probably because the people were interested in chicken development. This is why we have more. But when we say that we have a novel microRNA, it's really when we compare to all the microRNAs in all the possible species, not only in the chicken or in the pig.
I have another question. Is there a way to check for real targets for these microRNAs by looking at correlations between upregulation of microRNAs and downregulation of some potential target genes?
Yeah, that's what we want to do. So as Sylvain mentioned before, we would like to, of course, compute the correlation between expression of long genes and microRNAs. And if we find a very strong anti-correlation, then the gene could be a good candidate as a target for the microRNA. We also want to look at whether we find some special motifs in the through primary TR for the indicating the binding of the microRNA there. So there are several lines of evidence pointing to a possible relation between the microRNA and the gene.
Yeah, because actually it's one of the main questions regarding microRNAs. What's the real function of these microRNAs and what are the real genes that are regulated by them?
Yes, this is very complicated problem. Even in human, they have many false positives. But if we add several possible information, then we can hope that and also comparative genomics, because we expect to have conservation. So we hope that doing all this, we will get less false positives.
Thank you, Sarah. There was one question in the chat regarding the metadata, if I remember well, from...
Josana but replied actually. You see, this is
Yeah.
Claudia Casper, yeah.
Yes, we use the metadata that are provided by the Funk Data Portal. So if you go to the Funk Data Portals for submissions, you can get templates of metadata information, either for the samples, the experiments, or the analysis, and you have some examples that you can follow to fill the forms. And so by using this, you can get quite standardized metadata from different groups in the world or from different experiments within a project or different projects.
Herbie, for those participating online, I put the answer to the question in the chat with the links.
Okay. Yes, so perfect. Thank you.
Yes. I think we can actually stop and start again at 6:30 because.
Any questions in the room? Yeah.
There are some questions. Okay.
Is for you, Hervé. I think that you told us that you are also you are characterizing the other small RNAs that, or Sarah, I don't know, that you are expecting to characterize the other small RNAs generated in the.
So this is a question more for Sarah and Servan. But we have
Yes.
other types of small RNA, so let's go.
Yes, I think that in the protocol you use, you try to enrich for microRNAs, but that doesn't mean that we will not find other kinds of small non-coding RNAs, and we will characterize them as well. Yes.
And I have a question for Sylvain also about the integration of all the data that you are generating. Do you have some softwares or how to do the integration of all the data generated in the?
Now there are many methods. I hope you can hear me. So there are quite a few methods that are dedicated to integrative analysis, but those are, it's never like like a one click and then you get everything, because it requires a lot of steps of filtering and adapting the analysis to your specific data. Yeah, so it's a very active field of research, and we have quite a few methods that we are considering for this. The thing is, if we want to integrate everything we have, including correlation between gene expressions, anti-correlation with microRNA target predictions, ChIP-seq, chromatin accessibility, methylation, and the other one I forgot, but this is quite challenging, yes. And the size of the network you would get, because is very challenging. So usually these integrative approaches are not done at full scale. They do like transcription network, then they do integration of like, for instance, ChIP-seq and gene expression and stuff like this, in different steps. So we would like to adopt a more integrative, like a larger approach with by defining a network that would be scalable. And as Sarah mentioned, the trick is that if you run like microRNA target prediction, since the sequences are very short, you have a lot of false positive. So we need to, and it's a bit the same with promoter capture IC. We have many links. So we need to add the different layers to focus on the more like stringent signal. And the differential analysis, for instance, is something that we will use to focus on the most interesting biological signal, that the ones that are providing a significant switch. This is something we can extract assay per assay and then combine based on the results from the differential analysis. This is our best solution so far to explore.
Okay,
Is that okay?
Yeah, but the bottom line is that we would like to use the kind of knowledge graphs to do that, but and there are also some solution that exist like Neo4j and Cypher for the interrogation, and we are going to dig into that because it would be really nice to have all the data in the same graph rather than doing it like 2 assays per 2 assay. But it's not developed yet, but we are very interested in these approaches.
So anyone who's interested in joining the analysis team is welcome.
I have another question for Sarah. That you told us that you find the 45% of attack peaks in the intronic regions.
Mm-hmm.
Do you think that the result is because the annotation is not okay or fine?
No, I don't think so, because I think we find the same kind of number for human and mouse, which are much better annotated. So I think it's quite well known that you have many regulatory regions in the introns actually. Yeah.
Okay.
What will be interesting is to consider also the proportion of switching chromatin accessibility regions. I don't remember if we had this distribution, but I'm quite curious about the evolution of the distribution and distant regulatory sites that we find between like all of the ataxic peaks and the switching peaks. I think we have to compare this as well. I don't remember if we had them, but that's a good point.
The peaks were concentrated in the beginning of in the intronic regions of the beginning of the gene, or through all the genes.
This is a good question. I haven't looked into that. I consider all introns together, but it's true that we could split that into first intron or the rest. Good idea.
I have another question. So how do you find the switch for, you have a lot of switches from the gene expression and chromatin accessibility experiments. How do you find the switch? Did you put a log-fold change or something like that to discriminate big switch from small switch, or?
No. So far, this is just like significant results from differential expression or differential accessibility. So, which means that we have a p-value lower than 0.05. And this is everything that's significant. So, of course, this is why we have a lot of results also, is that something that has a very subtle variation in gene expression will be picked by this, but it's still significant, meaning that the 4 replicates in one stage have a lower expression, and the 4 others have a bit higher expression in a reproducible manner. So, but still, of course, we can discard some of those by increasing the threshold of fold change we want to consider. So that's one way to do. If you're, for instance, interested in some range of fold change, I know this is different. If you consider microRNAs, for instance, fold change, a very low level of variation can have a huge impact in biology, or messenger RNAs like coding RNAs. This is different. Yeah.
We have a question from Chris Tuggle, and I have one also just to finish. On gene ontology for the switch of the ataxic peak, do you see the same type of functions than the one you saw with gene expression, or you didn't do it?
No,
I think we did, right? No?
We did?
I don't remember.
No,
but
I don't
yeah,
think
it's in the list. Of course.
Okay.
The attack-seek, so of course, regarding the attack-seek peak function, we cannot infer so far all of the functions because we don't have the target genes so far of all the peaks, but the ones that are in the promoters, I think Servant did that maybe. The ones that are in the promoters, of course, we can look at the genes that are in those switching peaks and get the overlap with the genes that have the differential expression pattern. So yes, definitely something that we are considering for sure.
Chris?
Well, I had actually thought of a question just when you mentioned that. I mean, the other thing that people are doing is looking at correlation between changes in open chromatin accessibility and gene expression nearby. So that's another method for linking the enhancers with potential target genes. So presumably you're thinking about doing that as well. Yeah.
Yes.
Yeah,
this is,
absolutely.
yes.
Yeah, and we'd like to get a bit further into this direction, not just using the linear distance between the regulatory sites and the promoter, but also using the 3D distance that we get from promoter capture.
Right.
We are, yeah, we'd like to combine this information.
And then I put into the chat just a comment that I think, obviously, integrative analysis, if we can coordinate that, that would be very useful and learn from each other in terms of who's doing what types of integrative work.
Yeah, this is really true. This is a very very interesting topic, and it's an active field of research. So it's always very very good to communicate and exchange and share these methods and results. Definitely.
We have a bit of an opportunity for those who are going to PAG, and we have a slightly extended time for the FANG workshop on Tuesday. So maybe we can make that an important component of the discussion.
Any more questions? Okay, so on the chat. No? Okay, so we can have a small breaks before the plenary talk from Jack Dekers at 6:30.
Yes, precisely.
So in 9 minutes. So we will be back in 9 minutes. Thank you all
Thank you.
for.
Okay, so I present him. There are no people. Voglio dire, siamo veramente quattro gatti. Sì, va bene. Non so come mai. Questo cazzo di microfono, no? Cioè, adesso c'è. Madonna, ma sembra una strada. Mi viene meglio scriverlo. Okay, can I go? Okay, so we are back. And we are very honored to have with us Professor Jack Deckers, who actually doesn't need introduction, but I will try anyway. Jack has been professor of animal breeding at Iowa State University between 2002 and 2014. And since 2014, is Curtis Distinguished Professor in the Department of Animal Science, where he is also a section leader. Jack focuses his research on quantitative and statistical genetics, including the integration of quantitative and molecular genetics and genomics for QTL mapping, genome-wide association studies, and genome prediction and selection. And today he gives us a very interesting talk on the genetics and genomics of potential indicator traits for disease resilience of growth finishing pigs. Thank you very much, Jack, and I give you the floor.
Okay, thank you very much, Elisabetta, and yeah, thanks also for the invitation. It is indeed an honor to talk to this group. No, it's a keynote. It's called a keynote talk, and I think in part because it's not about FANG. This talk, although we are using some gene annotation information in some of our analyses, but hopefully it will still be of interest to many of you. Okay, so the talk is about resilience. Now, we know that when it comes to infectious diseases, resistance to some pathogens is controlled by a single gene, and the F18 E. coli mutation in that gene is quite famous for that. The mutation removes a receptor that in the intestinal wall that the bacteria uses to attach to the intestinal wall, so that the receptor isn't there anymore, or not functional anymore. Then the bacteria has no way to attach to the inner lining. But when we talk about other pathogens, most pathogens, resistance or susceptibility to most pathogens is highly polygenic, and maybe there are some genes with larger effects, and we've certainly seen that with infections or susceptibility and resistance to PRRS in pigs. But generally, it's very polygenic. Now, of course, there are examples where gene editing has been successfully used to introduce complete resistance of animals to a pathogen, and the PRRS mutation or the PRRS edit in the CD163 gene is a good example of that. We're here focusing on using existing genetic variation rather than creating new variation by gene editing. Not that that technology isn't very promising, it is, but is it going to work for all the other pathogens that are out there, where there may not be single or a limited number of genes that can be edited to make the animals resistant. Also, the pathogen is evolving, and there are new pathogens on the horizon, well, that we don't even know yet about yet. So for that reason, we have focused on disease resilience, which is the ability of an animal to be minimally affected by disease or to rapidly return to the state before the disease. So basically the ability to cope with disease, given that we're not going to be able to eliminate all pathogens from the environment, or make animals completely resistant to all pathogens. And that's, of course, complicated by the breeding structure that we have in livestock species. Here, an example in pigs, where the selection and most of the data recording takes place in nucleus herds that have to be maintained with high biosecurity. And that, of course, makes it very difficult to collect data related to disease resilience. So to address this, I've been fortunate enough to be a part of a large consortium led by Mike Dick and out of University of Alberta, and Graham Plastow has been very involved. Graham, I believe, is online. And in 2015, as part of, we obtained funding from Genome Canada and Genome Alberta and Genome Prairie initially to start a large-scale project on disease resilience in grow-finished pigs. Quite a number of institutes and universities involved, as you see here, and also very important, a very strong industry component to this project through PigGen Canada. PigGen Canada is a research consortium of pig breeding companies. When we started, it included seven breeding companies that are listed here. Two of them have since dropped out, so we're left with 5 industry partners. And these partners, they provided the animals that we are working with, as well as funding, as well as helped us with directing the research and focusing on implementation. So that started in 2015, and what it involved was at a test station in Quebec, a natural challenge was set up, which was initially in a barn that included a nursery and a finisher. Pigs from neighboring farms with health problems, known health problems, were brought in to seed the barn with a host of pathogens. And then to maintain that through a continuous flow system. So every 3 weeks, a new batch of pigs from one of the 7 industry partners was entered into this natural challenge facility, where they had contact with the older batch that was already infected, and that's how the infection pressure was maintained. Before these pigs went into the natural challenge facility, they went into a quarantine nursery, and where we took a lot of samples, and that's a lot of what I'll talk about is what we collected there and how we analyzed that. So in total, so this ran for about 7 years. In total, about 4,700 Yorkshire landrace weaner pigs. They were barrows from clean multiplier farms from the 7 companies. They were entered over the 7-year period. So the pigs, they were born in clean multiplier herds. Barrows are sort of a byproduct of the maternal line crossbred program of these breeding companies. And so they were transported to actually what I'll show here. The test station was just north of the St. Lawrence River. US is here, Quebec is here. And they first entered into a quarantine nursery, and then about 19 days later, they were transferred to this challenge facility, which consisted of a nursery and a finisher in the same airspace. So this is where they were challenged, and eventually, if they made it, they were sent to market, and we also got slaughter data. So there are lots of pathogens in this barn, and it wasn't consistent from time period to time period. Sometimes some pathogen disappeared from the barn, and it had to be reintroduced. So quite a number of viruses. Initially, we also had PCV2, and the pigs were not vaccinated for PCV2, but because the challenge was a bit too severe, fairly early on, vaccination of pigs for PCV2 was implemented before they got into the facility. And number of bacteria and some parasites. And the weekly mortality rates you see here for part of this 7-year period, it fluctuates a lot. And there was a very dedicated veterinary team that kept very close eye on making sure that it didn't get out of hand, but also to make sure that we maintained disease pressure. So different strategies were used to mitigate or to maintain more or less level a certain amount of disease pressure. But obviously, it's a very dynamic system. So under the challenge, we collected growth performance. We have daily feed intake, also daily water intake, morbidity in terms of health scores, mortality, and all the veterinary treatments were recorded. So these are what we call our resilience phenotypes. They were collected on all 4,700 of pigs. And they were all genotyped using a high-density SNP panel. This is an example of one of the batches. The red ones are the ones that died. Reasons of death, quite a range, but a lot of intestinal problems and respiratory problems, reflecting the pathogens that were in the barn. And the number of individual health treatments, there were some group treatments, but then the individual health treatments, some pigs didn't need any treatment, and others required 10 or more. So quite a bit of variation there also that we saw. The first question we had, well, given that this is not a controlled disease, it's very dynamic from batch to batch. Is the data we're going to get out of this even heritable? And so Austin Putz, who was a PhD student at the time, now works for Hypor and Hendrix Genetics. He analyzed the initial data and found that, it's quite heritable. So this average daily gain in the challenge nursery and in the finisher, very respectable heritability, similar to what you would see in a nucleus herd. Also the same for feed intake and FCR, same for carcass traits, and also the ones where we're mostly interested in, number of treatments per day and mortality in the challenge nursery and then across the nursery in the finisher. No, low heritability, but higher than what we would typically see in literature, where the incidence of disease would typically be quite a bit lower. So again, keep in mind that these are multiple diseases that animals are treated for, and so we lump everything together, and the same with mortality, and yet we obtain fairly reasonable heritabilities. Another PhD student analyzed a larger data set and also finds reasonable heritabilities, and this has all been published. And he also looked at drinking behavior, and also we found reasonable heritability there. And then he also looked at reaction norms, where he quantified the disease pressure, and then looked at G by E, and found some evidence of G by E depending on the level of disease pressure, which, as I mentioned, fluctuated over time. He also did a GWAS, and this has all been published. I'm just going to show you the GWAS for average daily gain in the challenge nursery. So that's where they're mostly affected. And we find quite a strong signal on MHC. Now, when you look at the QTL database, and you look at growth rate QTL, there's very few reports of MHC showing a QTL for growth rate. And that's because most of these studies are done in healthy environment. And you can see here that under challenge, MHC does become important for growth rate also under the challenge. One of the main reasons for doing this study is to develop these early indicators of resilience that potentially could be measured at the nucleus level where there's no disease. And so that was I'll talk most about that for the rest of this talk. So in the quarantine nursery, quite a number of indicated traits were investigated. And the requirements we have for a trait to be a good indicator trait for genetic selection is that it has to be heritable, and it also has to be genetically correlated to the resilience phenotypes. And we looked at CBC, natural antibodies, high immune response, in vitro immune test, phagocytosis test, and the blood transcriptome, as well as the proteome, metabolome, microbiome. And then finally, I'll talk about stress hormones in hair. And all of those were collected in the quarantine nursery, prior to the animals getting into the challenge. Now, CBC, I'm not going to talk about. Shoshun Bai, a PhD student with Graham Plastow, worked on that, and that was published. I also not going to talk about the blood proteome, that's also been published, nor about the blood metabolome. Elda Dervishi, a postdoc in Graham's lab, worked on that, and that has been published. Some very interesting results, but I just don't have the time to cover all of that. What I am going to talk about are the ones that are in red. So the first one is natural antibodies. Yulu Chen, a PhD student in my group, worked on that. Natural antibodies are present even in the absence of pathogens, and they bind to conserved antigens across pathogens. We looked at 4 of them, 4 of these mitogens, and or antibodies, natural antibodies to 4 of those mitogens, and that was all done on blood that was collected in the quarantine nursery. So again, before they entered the challenge. And she found reasonable heritabilities. She looked at 2 isotopes, IgG and IgM, natural antibodies to those antigens. IgG was lowly heritable. IgM was quite reasonably heritable. And IgG, we also had quite a large maternal effect, which is not surprising since IgG reaches the piglets through the colostrum, and they're only about 30 days old by the time these blood samples are taken. So in terms of genetic correlations with resilience, and this gets a little complicated, but just want you to focus on the trends. So this is the IgG and natural antibodies and their correlations with mortality in the nursery, in the challenge nursery, in the finisher, number of treatments, and also average daily gain in the quarantine nursery and in and under challenge, as well as feed intake. Here you see that KLH IgG and LPS IgG are positively correlated genetically with growth rate under challenge in the challenge nursery when they're first exposed to the pathogens. And there so the interpretation there is that higher levels of antibodies to those antigens, IgG antibodies, is genetically correlated with higher growth rate, so giving some protection or being associated with some protection. Standard errors are large, and that's one thing you'll see throughout these immune assays. We were not able to do on large numbers of animals, so typically about 1,000, a little under 1,000. And we just looked at lots of immune assays to identify ones that are promising that then can be further investigated. So also for mortality, you see negative genetic correlations, so higher level of that natural antibody genetically associated with lower mortality, but large standard errors. When we look at IgM, higher heritabilities, and most of these tend to be negative, but again, no large negative correlations and large standard errors. But there is some indication that these natural antibodies could be indicators upon further validation. So especially IgM, because the higher heritabilities, but genetic correlations definitely need more validation before that can be implemented. The other technology what we looked at was the high immune response technology, patented technology from Bonnie Millard at the University of Guelph that has been implemented in in cattle, in dairy cattle, and also beef cattle. So she measures cell-mediated immune response and antibody-mediated immune response. In dairy cattle, she finds heritability of about 30%. The cell-mediated is like a TB test where you inject an antigen and then you measure the skin thickness. I think 1 or 2 weeks later, and this is an ELISA test. And she has shown that these tests are genetically correlated with disease resistance, with disease incidence, and has been implemented by CMAX Canada as Immunity Plus. And so when we started this project, we approached, several of us approached Bonnie and said, well, can you also implement this in pigs? And so that was done. So Julie Schmidt, who is a postdoc in her lab, Bonnie's lab, so worked with the staff at CDPQ at the facility to implement these both tests. And here you can see that it's injected in the flank, and the other one gets a placebo, the other side, and then they measure thin thickness 1 or 2 weeks later. The heritability, so Mark Cheng, who was a PhD student with me, analyzed this data. He finds reasonable heritabilities, a little bit lower than they find in dairy cattle, and some litter effect for the antibody-mediated response. The genetic correlation between the two was slightly positive, so that there is some genetic correlation between these two arms of the immune system. What we are, of course, interested in is, well, if we measure that in the quarantine nursery, AMER and CMER, is it genetically correlated with subsequent disease resilience? So here you see growth rate in the quarantine nursery, in the challenge nursery, and in the finisher. Low correlations. They're a little bit larger when we look at the number of health treatments that the animals receive in the nursery. And also for mortality, they're a little bit larger, but large standard errors. What surprised us is that for CMER, we found an opposite direction of these genetic correlations than what we would have expected, and what's seen in dairy cattle. In dairy cattle, higher AMER and higher CMER is associated with less disease, and here we find that also for AMER, but not for CMER. And this may indicate that the pigs, although they came from clean multiplier farms, they may have been exposed to some minor pathogens when they showed a response that when we take the samples in the quarantine nursery. And we'll see some evidence of that coming back later on also. Mark also did a GWAS and found a very strong signal for MHC for AMER, but not for CMER. For CMER, it was very, very polygenic. The next immune assay that we looked at was proliferation of PBMCs from young healthy pigs. And again, those were done on blood that was taken at the quarantine nursery. The work was done at McGill University, Caroline Gilbert, and it's been published. And so we looked at a total of almost 900 pigs and exposed the PBMCs to 5 mitogens that are listed here, and then computed the blastogenic index score at 3 time points after stimulation of the PBMCs. And that's the results I'll show you. So heritabilities, so the 5 mitogens here, heritabilities at the different time points, and then also area under the curve, reasonable for CON-A and PHA, and also for PMA, but not for PWM and LPS, surprisingly. So in terms of genetic correlations with resilience, we only looked at the ones that are indicated, well, the 3 that showed reasonable heritabilities. So those and these results are also all published. The genetic correlations of response to these mitogens at the 3 time points with the number of health treatments in the challenge nursery, and given the large standard errors, it's most important just to look at the overall trends across time and across different resilience traits. And here they tended to be negative. So higher response to of the PBMCs to these mitogens, fewer treatments genetically. When we look at treatments across the challenge nursery and the finisher, correlations get a little bit lower, but still that same tendency. Mortality, also, even larger standard errors, but a tendency for the correlations to be negative, so less death, lower mortality, lower risk of mortality of pigs that genetically have higher response to these mitogens. Mortality in the finisher and across the nursery and the finisher. We also did health scores, so scoring pigs. Every pig is scored on a scale from 1 to 5, with 5 being more healthy. And all those correlations, again, huge standard errors, but all those correlations were quite large negative. Most of them were quite large in both the challenge nursery and the health score in the finisher. Growth rate tended to be positive. So pigs that had a better response, a better immune response, PBMCs responding higher, tended to have higher growth rate in the challenge nursery. And in the finisher was a little bit less clear. So, some interesting results that came out of this, but again, they need further validation to deal with the large standard errors. Next, we looked at immune stimulation assays on blood of young healthy pigs. So in contrast to the PBMCs, that needs to be done on fresh blood, isolating the PBMCs and stimulating them. These assays can be done on stored blood, and this work came from Claire Rogel-Gaillard and Fanny Blanc at INRAE. And also was actually most of the assays were done in Graham Plastow's lab. Fishesh and Shushun Bai analyzed the data. Shushun was a PhD student with Graham and now works for Turkey Hybrid, Hendricks, and Fishesh Bhatia is still a PhD student with me. And so they looked at a total of over 1,500 of these pigs, again, blood taken in the quarantine nursery. And they looked at both the concentration and the fluorescence activity of these 9 cytokines. And then under 3 conditions, non-stimulated blood and blood stimulated with LPS and a combination of PMA and ionomycin. Heritabilities were tended to be low for the non-stimulated assays, blood without stimulation, so the measurement of those cytokines in unstimulated blood. But with stimulation, especially LPS stimulation, we got slightly higher heritabilities. So that definitely was promising. In terms of genetic correlations, and this was only done for the more heritable cytokine levels and assays. And here I'm showing growth rate in the quarantine nursery, so without challenge and under challenge. So generally, these correlations are negative, indicating that pigs whose blood responded produced more cytokines after LPS stimulation, had lower growth rate in both the quarantine nursery and under challenge, which is maybe opposite to what we expect. I'm not quite sure how we can explain that, but that's something that we still are trying to investigate. And to some degree, we see the same for the PI stimulation and a little bit even in the non-stimulated. Okay. When we look at genetic correlations with treatment and mortality, they tended to be positive. If you look at the red and the gold, or the cardinal and gold, which is the treatment and mortality under challenge, where you see most of the disease, they tended to be positive. So again, opposite direction to what we maybe expect. They also looked at a phagocytosis test, which can also be done on blood samples. same group of people involved. This was done on almost 1,000 animals. Again, assays done on blood taken at the quarantine nursery, looking at 5 different cell types and both the cell count, the percent of those cells that phagocytize, and their fluorescence activity, so how much they phagocytize. Heritabilities were reasonable, up to 40% or 50%. The cell counts, but also the phagocytizing percent, fluorescence a little bit less heritable. Genetic correlations, we focus on the red one, that's growth rate under challenge. Fairly low for cell count, and Ashu Shunbai has a separate article where she showed that also, that the cell count measured on healthy animals isn't very predictive genetically of disease. Percent phagocytizing ability there was some evidence, but again, opposite direction to what we expect. So pigs that have a higher capacity for phagocytizing genetically have lower growth rate under challenge, although that did recover in the finisher. And also for fluorescence, it was a little bit less clear. There was something going on in the quarantine nursery with large standard error. So also that needs more investigation. And then with treatment and mortality, if you look at treatment, the cardinal color here, tendency for, well, this is with cell count, with phagocytizing ability. So here the cell count, higher granulocyte count, a tendency for genetically more treatments, but fewer in the for phagocytizing ability. Here we do see the expected direction, that higher phagocytizing ability, tendency for fewer treatments and less mortality. So, and especially the eosinophils are interesting here, although I understand that the eosinophils don't have a huge phagocytizing capacity, but we did find that negative genetic correlation in a favorable direction for both phagocytizing ability and fluorescence of eosinophils. Then we also looked at the blood transcriptome. So again, those were taken in the quarantine nursery. We used the QuantSeq or 3' RNA-seq with a globin block to analyze, to get gene expression on 1600, and we actually now have it on 2500. There's Kyu-Sung Lim, who was a postdoc in the lab, is now a faculty member in South Korea. He did the initial analysis at the phenotypic level between gene expression and the resilience phenotype. So is gene expression level measured on blood, is it predictive of disease resilience phenotypically? But here what I'll talk about is the genetic analysis that he also did, and that is about to be accepted by GSE. So he estimated heritabilities of all 14,000, the expression of all 14,000 genes in blood of these about 1,600 animals. And he did it with or without correction for CBC, for cell counts, because dealing with the fact that blood is a mixture of cells, and to account for cell composition. Generally, heritabilities with and without, they tended to be very similar. So not a huge effect of cell composition on heritability estimates. But for litter effects, we did find, and it's kind of expected, that expression of mitochondrial genes had large litter effects, and so also some genes on the X chromosome. And of course, we do an analysis where we don't account for mitochondrial DNA or the sex chromosome. So that's kind of what you would expect to see. And then you looked at genetic correlations with resilience, all the different traits here in the different phases of the challenge, and the number of significant genes, depending on whether it was with or without correction for CBC, or was significant for both analyses. So we estimated all these genetic correlations, and significance at the 0.05, but then when you correct for multiple testing, then FDR, there are very few genes that have an FDR less than 0.2. So still, even though we have 1,600 animals, it's still a limited data set when you want to get a genetic correlations, which is what we need. So what he did is he did a GSEA, gene set enrichment analysis on the genetic correlations. So here, either without or with correction for the CBC, all the traits on the different phases, and then looked at the genetic correlations of all the genes, expressed genes in blood with these traits. And then it did a gene set enrichment analysis based on Go terms. And he did find that there are multiple immune terms that come up that when you look at the genetic correlations. And the red here indicates that the genetic correlations of genes with those Go terms that were classified with those Go terms tended to be unfavorably correlated with disease resilience. So generally higher level of expression of those genes related to innate immunity, adaptive immunity, or both, tended to be higher expression of those genes tended to be associated with lower resilience. And again, remember, we're looking at a measurement of gene expression in the quarantine nursery when they're still fairly healthy, although this may also be a result of the fact that there may have been some disease pressure already even before they got to the quarantine nursery or in the quarantine nursery, although none of the major pathogens were detected there. So yeah, so when you look at cross genes, there is some evidence of associations between gene expression and disease. Too early to get into any indicator traits or indicator panels that we would develop with gene expression. We need to do more investigation to look into this. As you can see, for a lot of this, we've just only scratched the surface of a lot of the analysis that need to be done with these data. So now we have added another 850. So we have almost 2,500 animals with gene expression data in blood. And now with a grant from NIFA led by Chris Tuggle, we are doing, he's doing a lot of single cell RNA-seq in blood. And now the plan is to do deconvolution of the expression profiles to get expression profiles of individual cell types on all those animals. And that hopefully will tell us a bit more yet. The final thing I'll talk about is something that's also still ongoing, where we're looking at hair. Instead of blood, we're looking at hair. Of course, that's also an easy sample to collect. It's actually cut hair, not pulled hair. Because what happens when your hair grows, hormones that are in the bloodstream are deposited in the actual hair. And so as your hair grows, it reflects the hormone levels that are in the blood at the time of growth. So you could, in principle, take the tip, and that would represent stress hormone levels in blood during the time that part of the hair was grown. No, we're not splitting the hair. We are actually cutting the hair 3 times. The same patch on the back is cut when they just before they enter the challenge nursery. So then we're looking at hair that was grown since birth, basically, and looking at the effects of response to stressors that the animal was exposed to during that time, the stress of farrowing, weaning, being mixed, transportation in this case, changing diet. So a lot of stressors here. And then that same patch is cut twice. So here, where it would mainly reflect disease stress, and here also disease stress and potentially also other stressors. And the idea is that there's lots of stressors that animals are exposed to. In the disease challenge, we're mainly looking at immunological stressors, but all these other stressors we'll see here, and also partially also at the other 2 time points. And the hypothesis is that response to non-infectious stressors are genetically correlated to responses to infectious stressors, and that's one of the things we're getting at. But then, of course, we're also using this to see whether they are genetically correlated with disease resilience. And so that's the data we have so far is just on cortisol and dihydroepiandrosterone taken in hair at the quarantine nursery. And we are also doing the back test was done at that time point, almost 1,000 animals. Heritabilities of the back test were quite good. For cortisol, about 0.2. DHEA was not very heritable. Well, actually, at the 0 heritability, it did have some litter effects that you can see here. And then we also looked at the ratio. Genetic correlations of cortisol with the back test. The one reason we did the back test is to investigate, well, is what we're measuring in hair at that time point, is it genetically correlated with a standard stress response test? And we find that those genetic correlations are positive. He also, Fazir has done a GWAS on cortisol, and we find a very strong signal, more than 60% of the genetic variants that is associated in the window that has the NR3C1 gene, which is a gene that modulates the availability of cortisol and the HPA axis. So this is a very strong candidate that we are looking into further. That's all the work that we've done so far. Of course, now we're also going to get haircuts at the later time points. So we'll be able to correlate them with cortisol and other hormone levels that we are measured at this time point, and also looking at correlations with disease resilience, which is underway. So in conclusion, we think disease resilience is a useful target to improve host response to multiple pathogens and future pathogens that may come along. To do that, we established this natural challenge model. We found heritable variation in growth, feed intake, mortality, morbidity. We did lots of immune assays. Lots of results are still inconclusive. Needs more analysis and more data. The same with the transcriptome and some encouraging initial results for cortisol. And hopefully we can answer this question also, whether disease resilience parallels resilience to other stressors. So with that, I want to thank all the collaborators, and hopefully I've left some time for questions, but most of you will probably be hungry.
Thank you very much a lot for this talk. We cannot clap because anyway, but we can try.
Thank you.
Okay. Any question for Jack? Not yet? Okay.
Everybody's ready to go eat.
So, I mean, there was an incredible amount of information, and it's also amazing to see the numbers that you have in this study. So you are above 2,000 animals now that you can type and characterize for everything. And I underline that I really was pleased to have Jack to, because we didn't really tackle disease in the second part of GENE-SWitCH that we will have tomorrow, but will be really like that this talk is really a kind of a grasp already to the plan, the program that we have tomorrow, where I also hope Jack will join. So one thing that is really interesting is that, if I understood well, I mean, for example, when you look at your immune genes, you have, as a trend, if I understood, you have levels of immune genes that actually are anti-correlated to resilience, correct? I mean,
yeah,
you have high amounts, but in fact, high amounts don't correspond to more protection.
Yeah, we saw that with the cytokines and also phagocytizing ability, and not with the stimulation of the PBMCs. Yeah, what the reason behind that is, it's not clear, and it may be that, you know, that, and it may relate to the response to non-infectious stress also, which we know that immune system is involved in that, because these pigs, when we take most of these samples, they've just been transported across Canada. They're on a mixed. They're on a new diet. Blood samples are taken about 7 days after, and then also some of the assays were done on blood that was taken about 18 days after they arrive. But lots of things going on that may affect what we measure in blood at that time point, and how it could relate to disease resilience. So maybe they are already more stressed at that time point, and we see that in some of these immune assays, and they may be more susceptible to those stressors, as well as to infectious stressors. But that's all speculation at this point.
But you could actually probably test it with new measures, I guess. You could try to modulate it,
Yeah.
And and I don't know if there are questions. In the meantime, please think of it. Yes. Alan. Okay, so I give the floor to Alan because he's connected with a question with Jack. To Jack.
Hi, Jack. There's a great talk. There's a sort of long-running dogma that if as an animal you invest in your immune system, that compromises growth. Any evidence from this enormous body of work to support that dogma, or to knock it on the head?
Yeah, we haven't. It's a good question. We haven't looked at directly at that, but I mean, we do have some evidence that in the challenge nursery, there is that trade-off. But then we also have the pigs that make it into the finisher. They are sort of on a recovery phase. So the other thing that we also have to look at, maybe that the pigs that respond strongly to the immune challenge, they may have lower growth rate in the nursery, in the challenge nursery, but then they get over it and they actually grow faster in the finisher, right? Because there's this whole allocation theory. There's only so much energy and it has to be allocated. But another part of resource allocation is how flexible animals are in reallocating, right? So, and we've seen that in some of our PERS work, that the animals, actually the line selected for low RFI to be more efficient, you would think that it puts all its energy, it can't mount a very strong immune response. It actually mounted a very strong immune response, dealt with the disease, and then went on growing. Whereas the high RFI line, the inefficient line, didn't have as strong as an immune response, and as a result, the infections kind of lingered on, right? So this metabolic flexibility, as we call it, is another aspect that we have to look at. And but yeah, no, we haven't looked at that in great detail in these data.
Thanks.
Okay, so we'll give the floor to Maria Ballester, who has a question. She's in the room.
I have a question regarding the immunomarkers that you tell us that the correlations are opposite in the experiment about the cell-mediated and antibody-mediated immunoresponses, that you were expecting the same results that in cows. And my question is that if you have seen that there are a high proportion or the mortality is more related to respiratory diseases, maybe that the percentage was higher, these results maybe due to the specific pathogens that are circulating in the farm, and maybe are expecting, because maybe you have more peers or more, I don't know. My question is.
Yeah, that's a very good point, and we're actually now starting to separate out the mortality and treatment reasons, right? So we can specifically look at the relationships with mortality due to respiratory disease or treatments for respiratory problems, and And I think that will get at your question, but yeah, we don't have those results at this point yet. So for now, we've lumped everything together and looked at, regardless of reason, we looked at mortality and treatments. Yeah.
Thank you.
Okay. And there is another question from Peter Knapp. Peter Knapp is online. Peter? Well, he raised my hand, but Peter, are you there? Yes.
Yeah, yes, I'm online, but I couldn't unmute myself. Hi, Jack. Nice to see
Hello.
you again.
Yeah.
This is really good. You showed us about, I think, 250 genetic correlation estimates, and very, very few of them were statistically significant due to the huge standard errors they have. And most of them were based on something like 1,000 animals. Yeah? Now, what you could do, and maybe that would be a very interesting and useful little project, is push all that data into a power equation to figure out how many data we would need to make those estimates, let us say, robust and meaningful and trustworthy, etc. Any idea? Are you going to do anything like that? Because in 5 or 10 years from now, that may very well be the most important output from this little project. Know what we need to do to make it work.
Yeah, that's a good. We haven't done that, and I hadn't thought of doing that. I mean, of course, there is the main goal from an industry perspective was to identify potential candidates, indicators that look promising, and then collect more data on those. But yeah, I think we can get a better idea of how much we can certainly use our data to understand how much data we need. Yeah.
Yeah, because those potential candidates, from a negative point of view, a pessimistic point of view, that would take us back something like 15 years or so, when marker-assisted selection was supposed to save the world. And we all know what came out of that. So some kind of quantification of what kind of data do you really need to make this work in a meaningful way may be an extremely useful bit of information.
Yeah,
I would argue.
Yeah, no, that's you're correct. Yeah,
and you seem to be sitting on that kind of data.
Yeah, yeah.
Okay, thank you.
And yeah, the other thing that I mean, there's still lots more we can get out of this data. One of the things that we are doing also is now with all these immune assays. I mean, they're all correlated with each other also, right? And so that we measure different aspects of the immune system with different types of assays. And so one of the things we're also doing is trying to get more biological understanding of which aspects of the immune system are most important when we aggregate all the data together.
Yep. Okay. And to come back 4 minutes ago, when you said reallocation may be just a thing, I would very heartily agree with that. That is what we need to focus on.
Yeah.
Okay, thank you.
Yeah, thanks.
Ricardo, switch me off, please.
We have another question from Marco Bink in the room.
Hi, Jack. This is Marco Bink. And a bit follow up on what Peter just mentioned about how many animals do we need. I had a question about given all what you learned from this project, and to what extent is the choice of your material critical, and the way you organize it? Like you had the large white landrace crossbred, but they came from 7 different providers from different locations, etc. You mentioned that these animals may already have been stressed before starting the experiment. Can you give any recommendations or reflection about what could be done better or in the next project?
Yeah, well, first of all, I want to say, I mean, I want to emphasize that I'm part of a large team, right? And Graham was very involved and others. So I'm just the one speaking to you today. But so I didn't do all the organization. But in terms of what can be done better, I think it was important that we had multiple lines involved, animal lines, and that they were crossbred, because commercial animals are crossbred. And of course, always a challenge is to get access to those animals, and breeding companies were very eager to collaborate on this. They see it as pre-competitive research that is very important to all of them. But yeah, I mean, it's impossible to get nucleus animals, and so multiplier barrows from the Yorkshire Landrace cross were available, and so. But yeah, then multipliers aren't as clean as the nucleus. So whether that has a big impact, whether what we see on these animals in terms of their immune assays also applies to the purebred nucleus animals, we don't know. If you really want to look at implementation, then it would probably have been better to get nucleus animals for the immune assays, for the indicator traits, but we need to measure the disease resilience on crossbred animals, right? So it's not easy, but there are things that can be done to better target the specific questions that we have in terms of developing indicated traits that can be measured on purebred nucleus animals.
Thank you.
Okay, I think there is a question in the chat. So I'm trying to recover it.
Yeah, from Chris. Chung et al. Just published a paper that indicated RNA traits can explain a large proportion of heritability in cattle traits. Can you talk about the EQTL work and any evidence that these data can explain the resilience traits? Well, we've done it at the phenotypic level. We've tried to predict the disease resilience from the gene expression work, for the gene expression data, and we used the genomic prediction machinery as well as machine learning. And it's not very, at the phenotypic level, it's not very predictive. At the genetic level, well, the genetic correlations we're finding, they do indicate that that the gene expression in blood at this early age does explain at least some genetic variation of disease resilience. We haven't done the calculations to exactly say, to talk about how much. The EQTL, yeah, of course, when we do the EQTL work, that's regardless of the resilience data, right? We just look at analyzing, identifying QTL for gene expression. But then that may lead us to better candidate genes for disease resilience also. But we're not far enough along there yet to be able to say anything about how much that can explain.
I had also a more general question, Jack. It's about the durability of this facility that you are using, because if I understand, you started in 2015, and I can imagine the amount of permissions for ethics and all this that you have endured in all this. So, I mean, it's something that probably in Europe it would be extremely difficult to realize. And I was wondering, I mean, do you have a term line, or you think that you can just continue? I mean, you will continue for long, or you will stop? Have you planned to stop in this?
Well, again, it's not me, and it's not me making the decision, but it's the test station of CDPQ, so actually the Quebec government, provincial government, and the staff there. The main person is Frédéric Fortin, and they have actually now terminated the experiment because it's expensive, and we're not able to get additional funding to continue this work at this point, right? But yeah, I mean, it did involve a lot of certificates, health certificates, and approvals, and which, yeah, I agree, may be more difficult in Europe. And not to say that, in Canada and the US, it's everything is allowed, because there was a lot of oversight and a lot of going back and forth between the staff at CDPQ and John Harding, who's a veterinarian from the University of Saskatchewan, was very involved, and a whole team of veterinarians to put in place protocols to minimize the suffering, but still maintain a strong disease pressure so that we get data that's relevant.
Yeah. I didn't say that in the US is easier that much. It's just that, I mean, it was just an assumption from me.
Yeah.
So I don't know if there are other questions. Probably not. And in this case, Jack, thanks a lot again.
Okay. Thank you.
Really, thanks for being with us. And I hope you will be with us tomorrow as well.
Yeah, tomorrow not early, and I'm teaching tomorrow morning, so I may not be only the last part I may be able to attend of your meeting.
Yeah, but anyway, it will be a pleasure if you are there.
Okay. Thank you.
Thanks a lot.
Bye-bye.
And I think we stop for the day. And we see you tomorrow, everybody. Thank you for being here. I think everybody's gone.
Just logistic information. No, here for them. So we will have the dinner in the Uber room, which is towards the exit on the left, in 10 minutes. Well, we were supposed to start now, but I mean, I asked for 10 minutes today. So everyone, I hope everyone will join, and see you there.